A very good almost evening to all of you children. Thank you for waiting. There was a small technical issue. I hope it's sorted now. Everyone out there, children, say a hello. Let me know that you can hear me, you can see me. Is everything cool? I see a lot of children already up there. I'm so sorry for keeping you waited. Hello, hello, hello. Can you respond? Hi, ma'am. Okay. Jafina says hi, ma'am. Hi, Jafina. Rakshit, we'll talk about RCP later, okay? We'll make our later on. Chinmay, hi. Zamir, hi. Gauri, hello. Liza, hello. Ashwini, hello. Rahul Rathod, hi, dear. Baswaraj, Ashraf, Roshan, Neil, GS World, Bhumika, Suresh, Ashraf. Okay, lots of children are there. So, ma'am, cover all the second PU chapters. So, let's start. Hi, Queen. Okay. Now, children, so what is the motto of this particular video? So I am going to solve 140 plus questions, and this is going to help you score more than 45. But can we do it in one stretch? No, which is why it will be there in parts, part 1 and part 2. Part 1, I would be focusing on these chapters which are of very high priority, that is sexual reproduction in flowering plants, molecular base of inheritance, principle of inheritance, organisms and population we'll do it in next part, okay? Human health and diseases, human reproduction and biotechnology principle and processes. So instead of organism and population today, I would be taking up reproductive health because it would be comparatively easy for you to understand things. If you study all of the chapters together, it will be difficult, okay? I am hoping that I am very much audible now. Roshan, is the voice fine? Okay, Chinmay, even I am very glad to see you all over here. Liza, voice is low. For everyone else, is the voice fine? If it's fine, just give me a heart or just give me a thumbs up. It will be really helping me well. If it's fine. Heart. Okay, Rakshit said, Rakshit gave the heart and Ashraf and Akshita. Okay, chalo, let's start. For those of you who are saying the voice is low, can you just put your earphones once more and try it up? It will be really good. Chalo, let's start with the first chapter, principles of inheritance and variation. So the rule is, I show you the question, you try the question, you tell the answer like 1, A, B, C, D, whatever is the answer. Chalo, let's begin. First one, select one word for the statement. If F1, okay, F1 resembled both the parents. If F1, my dear children, resembled both the parents, what is it known as? You have to choose from here. If F1 did not resemble either of the two parents, what is that? If F1 resembled either of the two parents, what is that? So give me the answer like 1A, B, C, D. 1A, B, C, D. If F resembled both the parents means both of them are equally dominant, right? Both of them are equally dominant. What do you call this condition as? 1A, okay. AK Volleyball says 1A, Chinmay says D, Ashwini says A. So I see D and A options are the ones that you're getting confused with. So let's see. So over here, listen, they have made everything in wrong order to confuse you guys. So you have to be really careful. So A is, if F1 resembled both the parents, that means A is codominance. So where is A codominance in both these options? So I can actually cut off these two options. Now B, if F1 did not resemble either of the two parents, that means it is what? Incomplete dominance. So B should be incomplete dominance. And where do you find that? Over here. Okay, now we are left with D. So C, that is if F1 resembled either of the two parents. For example, you have capital T and small t. If tallness is being expressed, this is what? Complete dominance. This is what? Dominance. So C is dominance. So with that, I'll say D is the correct answer. Yes, this is how things get messed up. Okay, just let me put on the AC. Yes. AK Volleyball, ma'am, I'm so sweet. Thank you so much, dear. Okay. Mine's correct, Queen. Every one of you who got the answer correct, kudos to children. Chalo. Let's move ahead with the next question. Are we ready? Are we ready? I'll read out all the names, okay? You read out the question first. So, read out this question. Which of the following is related to hemophilia? You have to tell me the answer and then I'll be reading out the names of children who got the answers correct. Or just who attempted at least. Okay, so I see Akshita, Ghost, Chinmay, Imran, Preeti, Basuraj, Rubina, Akshita, Kirtana, Monisha, Kach, Yashashwini, Zamir, Gauri, Jafina, Rakshit, Muhammad Ashraf, Supriya, Bhumika, oh my god, Megha Reddy, GMR, Shashank, Chinmay, lots of people are there. Queen, Subhashini, Suresh, GS World. Okay, now let's see for 2. So it says that you have to look for hemophilia. A recessive gene responsible present in the X chromosome, a dominant gene responsible present in the autosomal chromosome, okay? A responsible dominant gene present in the Y chromosome, a responsible dominant gene present in the autosomal chromosome. So my dear children, first thing, hemophilia is what type of disease? Sex-linked recessive disorder. So over here, hemophilia gene, the gene which mutation of which causes hemophilia is there on the X chromosome, which is why it is known as sex-linked disease, yeah? And the condition is recessive gene disorder. That means, for example, if a woman has 2 X chromosome, ma'am, and 1 is affected, will she have this disease? No. It has to be in a recessive condition. That means both the genes should be affected. This is recessive disorder. In the case of male, since they do not have any other X chromosome, if at least 1 is affected, that means this person will be having it. I hope that is clear. Yeah, it's not dominant, it's recessive. Let's give a thumbs up for that. Everyone who got A, my dear champions, you are doing an awesome work out there. Let's go ahead to the next question and I'll read out the names of children who got it correct. Sickle cell anemia is caused by substitution of valine by glutamic acid in the beta globin chain of hemoglobin, caused by a change in single base pair of DNA, characterized by elongated sickle-like RBCs with a nucleus, and autosome-linked dominant trait. You have to tell me the answer for this. How do you answer this? So I see someone saying A, B, C, D. Do not say like that. Give me the answer in numbers. 3, A, B, C, D. Otherwise, I'll say your answer is correct, but you might be answering for second question. So you'll get your answer wrong. So just to avoid that, my dear children, if you can, tell the answer like this. You know what? The AC is not working. Why is the AC not working? Now it will work. Oh my God, I see so many people out there answering the question. So I see most of you saying A is the correct answer. Are you sure? Are you sure it's A? C, B. Okay, Pooja Kagal says B. Shardhamma Sharu says B. Shobha Murthy says A. Okay, Arpita says B. So, children, actually this is not the answer. This would be the answer. This is not the answer. This is what you usually pick. Okay, this is why I colored it. Why it is not the answer? Sickle cell anemia is when you have elongated sickle-like shape. For sure, that is correct. In the RBC, that is also correct. But does the RBC have a nucleus, my dear children? Erythrocytes does not have a nucleus. So this is a wrong statement. Until this portion it is correct, but with a nucleus, it becomes wrong. Now, why it is not A? It is caused by substitution of valine by glutamic acid. Wrong. Initially, you had glutamic acid. This glutamic acid is substituted by valine, my dear children. Okay, it is substituted by valine. So here it is written valine by glutamic acid. That will be wrong. Okay, always remember G comes before V. So initially you had glutamic acid and then came valine. So what was the codon for this? It was initially GAG, but this got replaced and it was GUG, my dear children. A comes before U, you can remember like that as well. Okay, does it work? Okay, now over here, an autosome linked dominant trait. Is that correct? An autosome linked dominant trait. That is also wrong because it is what? A recessive disorder. Yes. Now let me see who all got it correct. So children, hello Rohit, namaste. So all of you who got B as the correct answer, you are going great. Okay. Okay, so your B is the correct answer. It is autosome linked recessive disorder, not dominant. Fine. Chalo. Let's go ahead with the next question. Fourth question. Which of the following condition correctly describes the manner of determining the sex in the given example? Correctly describe. You have to find what is correct over here. So someone is saying, ma'am, explain B option. Explain B option. B option says it is caused by change in single base pair. So as you can see over here, initially you had A, but it is replaced by U. There is only single base pair change. Not all of them got changed. So it is the actual one. Okay. Yeah, children. Can you try attempting this question? Ma'am, my voice is not clear. Is it clear now? Which one of the following condition correctly describes the manner of determining the sex in the given example? XO type of sex chromosome determine male sex in grasshopper. Is it correct? Okay. Homozygous sex chromosome XX produces male in Drosophila. Homozygous sex chromosome determine female sex in birds. XO condition in humans lead to Turner syndrome determining the male sex. What is the answer? What is the answer for this? 4A. I see children saying 4A. Okay, kudos to you. Pallavi, GS World, Akshita, Rakshit, Shobha, Nitesh Gowda. Please not be sad if I'm not telling your name, okay? Aisha, Meera, all of you, my God, you are doing great. Okay, so children, over here, XO type of, that means there is one sex chromosome, but there is no other sex chromosome. In this condition, in the case of grasshoppers, it becomes a male. It is correct. Now, homozygous sex chromosome XX produces male in Drosophila? No. Drosophila and human being is same. Okay, it's same. So, in Drosophila also, XY is for male and humans also XY is for male, not XX. So, this is wrong. Now, homozygous sex chromosome ZZ determine female sex in birds? No. Instead of that, what determines the female sex in the case of birds? Birds show female heterogamity. That means only once birds are having something that is showing female heterogamity, as in females are going to produce two type of gametes, not one. So it will be producing gamete with Z and it will be producing gametes with W. So it is ZW for females. It's the only time females are going to show heterogamety. Okay. Now D. XO condition in humans lead to Turner syndrome determining male sex. XO condition is basically when you get one X chromosome from either the parent and you not get any chromosome from other parent. Okay, so this is XO condition. In this case, it should be a female. It should be a female but have masculine character. So it is not determining male sex. This would be wrong. Okay, it's a female only but have male character. Okay, is it clear? Yes, Shobha Murthy, you're absolutely correct with that. So A is your answer. Who all got A? Kudos to all of you. Who all didn't get A, it's totally fine. Things happen. This is how we rectify. And I hope you guys are understanding the concepts behind all the questions that we are doing, right? Yes? Okay. Ma'am, surely, Baswaraj is saying surely 40 marks, you can get 40 plus marks. So, yes, it will be good, okay? Let's be hopeful about that. Now, question number 5. Okay. Ma'am, please explain C option. Explain C option 4. Okay. Homozygous sex chromosome determines female sex. It is not homozygous sex chromosome ZZ, but in females it is ZW. ZZ is for males. Okay, if Z and Z comes, it is a male. In the case of female, it should be ZW. That is the thing. So this is wrong. Cool. Going ahead to the next question. In a dihybrid Punnett square, how many non-parental phenotypic combinations are present? Okay, PU chapters we are going to take in the next section, part 2. Okay, I initially showed the number of questions, number of chapters that I'm going to cover in this video, okay? Shobha, I hope you got it. Okay, so lots of people have started answering 5B, 5C, let's see what look at it. In a dihybrid Punnett square, my dear children, non-parental. So basically, suppose dihybrid means you have to take two characters. Suppose I am taking round and yellow, you know both of them are dominant, usually, right? And then you have wrinkled and green. Wrinkled and green. So when I take the F1, all of them will be round and yellow. When I take the F2, I get 4 different type of phenotypes. You might have studied about that. Round and yellow, wrinkled and green, and in between you have round with not yellow but with green, round and green, and then you have wrinkled, not with green this time, it will be yellow. So 9:3:3:1 ratio you have seen, no? So 9 of them will be round and yellow, 3 of them will be round and green, 3 of them will be wrinkled and yellow, and 1 of them will be wrinkled and green. Always remember the dominant parent you took initially would be having the highest number, and the recessive parent you took initially would be having the lowest number. Now over here 9 plus 3 plus 3 plus 1 is equals to 16. So out of 16, how many of them are non-parental? Something that is new. Something that is new. So here this is something that is new, round and green and wrinkled and yellow. 3 plus 3, 6. So 6 out of 16 becomes your answer for this question. Okay, so A is the answer. Who all got A? Shanta, Nigesh. Nigesh, check Baba, check it once. Okay. I hope you got it correct, my dear children. Ma'am, can I score 45 by watching your video? I am actually hopeful you would be getting that, more than that actually, but you have to make your concepts really strong for that. That also I want to say. Okay? Chalo. A woman has a hemophilic brother and normal parents. What is the possibility of her children having the disease? Now when this type of question comes, you have to take a pen and start doing. Ajay says, ma'am, it's raining in Carver, ma'am, and also lightning, but we love to see your class in this situation for our future. Oh, that's actually great, but be safe. Yes. Okay. Children over here, someone has started answering the question also. Srinivas, Chinmay, in Bangalore also it's raining. Yes, I am in the studio, so I do not know. Manju, yes. So, woman has hemophilic brother. For a brother to be hemophilic, my dear children, I told you it should be this condition. For a man to be hemophilic, it should be like this. Fine. And for a man to be like this, my dear children, where is he going to receive the X chromosome from? Not from the papa, not from the papa, but from the mother. So the mother probably might have given this X chromosome. Okay. Now, going ahead, what is the possibility of her children, as in the sister's children? Okay. Sister's children having the disease if she marries a man who has no history of hemophilia. Now, suppose the condition is like this. Okay, suppose the condition is like this, my dear children, and I am taking over here XH from the mother and X from the father. Okay, similarly, I can take this X and this X from the father. So, in that case, she is normal. In this case, she is a carrier. She is a carrier. Now, going ahead, if she marries a man who has no history of hemophilia, that means she is marrying a normal man. Okay, then suppose it is like this. I am taking the carrier daughter. Okay, I am taking the carrier daughter for knowing the exact number. So, over here, X chromosome, XH over here in this gamete, X in this gamete, Y in this gamete. This and this goes, I get XX, she is absolutely normal. This and this goes together, XY, he is absolutely normal. This and this goes together, she is a carrier. This and this goes together, he is a diseased person of hemophilia. Now, all are hemophilic, wrong, because all are not. 50% of the total generation is hemophilic. 50%, do you think? There are 4 children, out of them 2 are affected. But does she have hemophilia, my dear children? No. She is what? She is just a carrier. So, I cannot count it in the 50%. Now, 50% of the son may be hemophilic. If you see, this is a son and this is a son. Only this is affected. Only half of the sons are affected. So, 50% of the son may be hemophilic. This would be the correct option. Okay, so what is the answer? 6C is the answer. Every one of you who watching this video, are you getting this? Do you want me to explain it again? Shubhash, thank you so much, dear. Okay, can we go ahead? GMR, Aisha, Rakshit, Pallavi, all of you doing great, my dear children. Liza, thank you so much. So, I suppose you are understanding. Going ahead with the 7th question. No, ma'am, explain it again. Okay, I'll explain it again. Children, please wait for a second. I'll explain and I'll be coming back because it is important for everyone to understand, right? So, listen, a woman has hemophilic brother. So, a person male to be hemophilic, he should be having a condition like this. Now, when a male child is being born, obviously one chromosome he will get it from his mother, and one from his father. So, he receives either of them, but it cannot be X. He cannot receive X from the father. So, it will be from the mother, and if he is hemophilic, my dear children, at least the mother should be a carrier, at least a carrier, I am telling you. Okay, suppose she is a carrier, my dear children, this is what you get for the son. Now, what about the daughter? For the daughter again, it can be X and this X, okay, or it can be XH and this X. So, she can be a carrier or she can be normal. Now, I would take the worst condition. I will take the condition to be carrier because I want to see over here, right? I want to see the extreme situations over here. So, that is why I will take this condition where she is a carrier, and I am going to fuse her with what? If she marries a man who has no history of hemophilia. If she marries a man who has no history of hemophilia, that means it's just X and Y, right? Normal man. Now, over here, in one gamete this, in one gamete this, in one gamete this, in one gamete this. Suppose this and this comes together, you get this. Here, since both the chromosomes are not affected, she will be just a carrier. Okay, she is not having sickle cell. Sorry, she is not having hemophilia. Now, this and this comes together, it will be XHY. Here, in the case of male, he will be affected. Now, This comes together, it's normal, she is a normal child. This and this comes together, he is a normal male child as well. Now over here, my dear children, if you see, all are hemophilic, no, because these two guys are normal and she is just a carrier, okay. 50% of total generation is hemophilic, again no, why? Because she is just a carrier, he has hemophilia, just him, 25% has hemophilia. Now 50% of sons may be hemophilic, yes my dear children, why do you say so? This is a son, this is a son, after that only one is affected, so 50% of the son, right? And none of the above, which is why C becomes your answer. Okay, got it? I hope you got it. Chalo. Next, question number 7. The inheritance pattern of a gene over generations among humans is studied by the pedigree analysis. This is equivalent to what? This is equivalent to what? Can you tell me the answer for 7th? Okay, someone is asking Moinili Jasmina. What's my full name? My full name is Jasmine Joy, my dear children. Sometimes both are hemophilic. In that condition, both are hemophilic as in, what are you talking about? Both the parents? In that condition, everyone will be hemophilic, the children. So 7C, 7B, 7D, it's an easy question, my dear children. When you're talking about pedigree analysis, this is equivalent to what? Quantitative trait, Mendelian trait, polygenic trait, maternal trait. This is direct question from your NCERT, my dear children. It is Mendelian trait. It is what? Mendelian trait. All the genes, okay. So any effect in your genes, my dear children, it can be inherited. And whatever is inherited is being studied in pedigree analysis, which is why it will be Mendelian trait. So B is the answer for this. Let's go ahead. I hope that's clear. Question number 8. Question number 8. Pick out the correct statement. Hemophilia is a sex-linked recessive character. You have to find the correct statements, okay? You have to find the correct statement. Ma'am, explain once again. Which question, dear? 7th, or can you tell me? Ashwini, what's your full name? My name, Queena. Who's Queen? Okay, some other Queen is there. Okay, sorry. Pick out the correct statement. Hemophilia is a sex-linked recessive character. Down syndrome is due to aneuploidy. What do you mean by aneuploidy? Aneuploidy means either addition or deletion of chromosome. Okay, is Down syndrome caused because of that? Yes, my dear children. In Down syndrome, you get one extra chromosome. Which one? 21st chromosome. So yes, this is what? This is known as trisomy of 21st chromosome. So initially you got one from your one parent, another from another parent, but you're getting one more unknowingly from one parent. So it's basically three chromosomes. Okay, instead of having 3 chromosomes. This is known as trisomy of 21st chromosome. Now, phenylketonuria is an autosomal dominant gene disorder. Is that true or false? Is that true or false? It is absolutely false. It is autosomal recessive disorder. Wrong. Phenylketonuria is an autosomal recessive disorder. Correct. So this is correct. This is correct. Now, sickle cell anemia is an X-linked recessive gene disorder. Wrong. It is an autosomal recessive disorder. So this is also wrong. So what are correct? A, B and D. And where do you find that option? In A. And all of you said A is the correct answer. Kudos to all of you. You're doing absolutely great. Okay. Shall we go ahead? Malayali Zonde. Question number 9. Select the correct basis of DNA, RNA and amino acid of beta chain resulting in sickle cell anemia. This is a difficult question. Try to think for once. Okay, try to think for once. Select the correct basis of DNA, RNA and amino acid of beta chain resulting in sickle cell anemia. As in, you're talking about the mutation and things like that. Okay. Murtuza, my name is Jasmine Joy. Chalo. Children, we will talk later about where you guys are from. Okay. Now your KCET is there within 2 days. We have to be pretty serious. We will talk. Okay. Chalo. Come on, come on, come on, come on. So D, 9B, 9B, 9C. I see 9C. We need full explanation, ma'am. I'm giving full explanation, dear. 9B, 9B. Okay. Listen. So I see B and C, C and B things. When I say that sickle cell anemia is caused because there was GAG initially and it is replaced by GUG, right? And this changes the situation from glutamic acid to valine, right? Now over here, this is what, this is, as you can see over here, this is, you're talking about an RNA, GUG as in you're talking about RNA, right? But in the DNA also, probably there might have been changes, which is why you got GUG. In the DNA itself, you might have some changes, which is why you got GUG, my dear children. So we have to look for, initially it should have been this, so we are going to look for the DNA. Okay, since the DNA column is also there. So if it was GAG, then what would have been the DNA, my dear children? DNA would have been C, A goes with T, and this goes with C. It should have been CTC, right? It would have been CTC. Now over here, okay, over here, after mutation it became GUG, and after mutation it was coding for what? Valine. So now what will be your answer for this question? Can you try to find out? What will be your answer for this question? You already saw, right? You already saw. It would be what? This. And you may ask, how ma'am, how can we be? So basically over here, my dear children, this itself has got changed. This DNA itself has got changed over here. There is a base pair change, which is why instead of T, what you have to get? Instead of T, it had become A. So now what is the RNA it is going to code for? GUG, and GUG is something that codes for valine. You get it? Which is why your answer would be B. It is not C. How many chapters in part A? Why is C not the answer? This is what I said. So initially when you are taking CTC as the DNA, okay, it will make an RNA which is like if you are having if you are having CTC, then it will be GAG. It will be what? Coding for the normal thing, that is GAG. But we are talking about the mutated version. We want this thing because this is the one thing that codes for valine. So instead of here, if you want to make it U, what you should be needing over here? U goes with A, right? So it should be CAC, which is why this becomes the answer. Okay, I hope all of you didn't understand, might have understood now. Okay, Priyadarshini, how many chapters are there in part 1? You have these many chapters, except organism and population, all the other chapters, all the high weightage chapters are there. Works? Chalo. Now, coming ahead with the question that we were solving. You didn't understand? I didn't understand. Okay, explain it again. I'll explain it again. Listen to me carefully this time, okay? So, my dear children, our aim, what is our aim? Initially, it was GAG. Then we said it is GUG, and GUG is something that codes for valine, and GAG is something that codes for glutamic acid. After mutation, there is some changes. It has become like this. So we can forget this part. We can forget this part. We have to make a complementary DNA to this, okay? Because we want this mutation. So what is the complementary DNA to this? G goes with C, U goes with A, and G goes with C. This is the complementary DNA that we are gonna have. And in the DNA there was single base mutation, which is why initially it was something else. Initially it was T, but it is replaced by A, my dear children, which is why in RNA also it has become like this, coding for valine. So it will be CAC, GUG and valine. Okay? Works? Priyadarshini, children, is the voice okay? Others, is the voice okay? Can I have understood? Okay, fine. I'll go ahead. I hope the voice is also fine. Thank you, Priyadarshini. Chalo. All good. Chalo. Question number 10. Mother and father of a person with O blood group. Listen, blood group is very important concept. Okay. Mother and father of a person with O blood group have A and B blood group. Have A and B blood group. And the child is what? O blood group. What would be the genotype of both mother and the father? Listen, O blood group, for that to come, what do you need? You need 2 O's. You need 2 O's, then only it can come, because O is the recessive character. So if you need 2 O, obviously you have to take it from here. So the mother should be having 1 O gene and the father should also having 1 O gene. But they are A blood group, they are B blood group, right? So for this person to be A, I need 1 gene for A. For this person to be B, I need 1 gene to be B, which is why over here, my dear children, A is dominant over O, B is dominant over O, which is why this is B blood group, this is A blood group. But when this and this come together, you get O blood group. Now you tell me, what do you call this condition as? Is it heterozygous A or homozygous A? It is heterozygous A. Homozygous A will look like this. It is not same, it is heterozygous A. So mother is heterozygous for A blood group and father is heterozygous for B blood group. So who all got A as the answer? You are absolutely correct. Okay. Dear children, if you got C as the answer, let's see what you are saying. Both mother and father are heterozygous for A and B groups respectively. Both, oh, that is also correct. Both mother and father are heterozygous for A and B group. That is also same thing, right? Yes, that is also same thing. Means the same. Yes. So either way, it would have been correct, A or B. I think that's a typo error somewhere. Okay, children. So please consider this to be the first statement which is there. You can go ahead with that. Works. Now, going ahead with C is also correct. C is also correct. I think there was a typo error, which is why it is not. So both of them would be correct because both of them means the same thing. Both mother and father are heterozygous for A and B blood groups respectively. Mother is heterozygous for A and this is also same thing. So both of them are going to be correct. Now my dear children, listen to me carefully. Okay. Over here in this particular chapter, what was important molecular base of inheritance that you have to look forward for? First thing, you have to look forward for blood group. Okay. Second thing, you have to look forward for diseases. Okay, you have two sex-linked recessive disorders. What are those sex-linked recessive disorders? One is hemophilia and the other one is color blindness. In both these conditions, since it is a recessive disorder, males are going to be affected more. Males are going to be affected more. Then you have three disorders which are autosomal recessive disorder, which is sickle cell anemia, thalassemia and phenylketonuria. Know about all these diseases. Then you have what? Turner syndrome, Down syndrome and what do you call that? Klinefelter syndrome as an example for aneuploidy. Go through that once. Very, very important. Okay. Apart from that, what is a test cross? Anyone over here who know what is a test cross? Can you tell me? So over here I will be making you understand certain concepts also. It becomes easy for you to get it in your head. Okay. Can you tell me what do you mean by? Can you repeat? Aisha Mira. What do you want me to repeat, dear? So hemophilia and color blindness, they are sex-linked recessive disorders. We have sickle cell anemia, we have phenylketonuria and we have thalassemia. All of them are autosomal recessive disorder. Okay, all of them are recessive. Whatever is written in your book is recessive. Okay, then in one of the pictures, okay, in one of the pictures when you get it, okay, one of the pictures below it, autosomal dominant disorder is mentioned. What is the example of autosomal dominant disorder? Autosomal dominant disorder example is muscular dystrophy. Muscular dystrophy. None of the other places it is written. Only under the image, under the pedigree chart it is given. So please go through it once. Okay. And apart from that, you have certain aneuploidy condition. As in, for example, when you gain one extra chromosome, Down syndrome, 21st chromosome. Know the symptoms of Down syndrome. They are short-statured, palm creases there, mentally they are retarded and things like that. Klinefelter syndrome, okay, where you have an extra X chromosome. Turner syndrome, where you do not have an X chromosome. Now, Klinefelters, it is usually male, but it becomes, they have a feminine character because they have an extra X. Turner syndrome, they are usually females, but they have masculine character because they lack an X. So please know that, okay. And wait. Test cross, someone is saying, talking about test cross. So test cross is basically the cross between F1 to the recessive parent or cross between TT and TT. Wait. Okay. Test cross. The cross between F1 and the recessive parent is known as test cross. My dear children, actually F1, F2 does not play a big role over here. Actually, what plays a major role is, okay, when you have a dominant plant and you want to check whether it is homozygous or whether it is heterozygous. So for understanding that, you cross it with a recessive parent. For example, you want to know whether it's like this or like this. So for understanding that, you are going to cross with recessive parent, that is small t. This is known as test cross. Now all test cross are back cross because every time we are going back to the parent, but not all back cross are test cross. For example, I just want to randomly cross this with this. Will I call this test cross? No, this is just a back cross. Okay. You are not able to listen me? Hello, hello, hello. Guys, is the audio very bad or is it fine now? Sound is less. For all of you? All good, ma'am, no problem. Some were saying, Chinmay, sorry, can you put it again, dear? So it's going like this, I cannot actually see it. Fine, fine. So some children are saying it's fine. Can you please reload, please, once? For once, can you reload? It will be really good. Okay, now genotype and phenotype are same, 1:2:1. Yes, in the case of Snapdragon. Okay, in the case of incomplete dominance, my dear children, genotype and phenotypic ratio is the same. So when you cross a red plant with a white plant, all of them, when you see, they will be pink in color. They will be what? Pink in color. So in F1 generation, you do not have anything to say. But in F2 generation, when you cross them together, my dear children, you have capital R, you have capital R, smaller, and smaller. So over here, one is having capital R, genotype, so 1. Two of them capital R, smaller, so 2. And one of them smaller, so 1. So this is a genotypic ratio. What about the phenotypic ratio? This looks red, only one looks red. Two of them look pink, so 2. And one of them are white. So 1:2:1. Okay? Fine. Mohammed, okay, everyone is saying the voices. Okay, I'll ask my team to check also. Just a second, okay? Just give me a second. I have asked the team to tell me. Okay. So children, I told you about all these things you should be having in your head, and also pleiotropy and polygenic trait. What do you mean by polygenic trait? A polygenic trait is basically when you have multiple genes. You have multiple genes affecting one particular character. For example, skin color. And also height. So there are multiple genes affecting one particular character. Okay, that is polygenic trait. Now what is pleiotropy? Can you tell me what is pleiotropy? Yes, so children, I got the thumbs up from my team. They are saying it's audible, so please can you close the video once and try to rejoin the video? It will be really great. So I don't think it should be a problem. Some background noise. I don't know. No problem. So Murtuza is saying there is no problem. Others are saying, so please children, once. For once, can you reload it? It would be really, really great. Or I will hold it like this. Okay. Now, so that is done. Now going ahead with the next chapter, that is molecular basis of inheritance, my dear children. So can you tell me the answer for the question that I asked? What do you mean by pleiotropy? One gene specify more character. Exactly, Chinmay. Okay, exactly, Preeti. All of you are doing great. One trait controlled by 2 or more genes, that is polygyny. Pleiotropy basically means one particular gene is going to have 2 character. Okay, so you have phenylketonuria as a best example. In phenylketonuria, you have an enzyme phenylalanine hydroxylase which gets mutated. Okay, this enzyme is not produced, which is why what will not happen? There is a conversion of phenylalanine to tyrosine. That does not happen. This does not happen. And also one more thing does not happen. Since this tyrosine is not being produced, my dear children, tyrosine is very much important for the production of melanin that gives the skin color and hair color. So which is why your skin and hair pigmentation also gets reduced. So in a way, isn't it controlling 2 characters? Yes. So this is an example of pleiotropy. Okay, it is seen in phenylketonuria. Cool? Fine. Ajay, I will be coming to your question. When we go chapter wise, I will be coming to the question. Okay? Works? Yes, and also sickle cell anemia, starch granule. Yeah, that is also correct. Going ahead with molecular basis of inheritance, my dear children, question number 11th. Try to solve it. The result of the following reaction experiment carried out by Avery on Streptococcus pneumonia has proven conclusively that DNA is the genetic material. So what was that particular thing? Can you try to answer? And all of you who over here are talking to each other. My dear children, what is your goal? What is your goal? Is your goal to talk over here and waste your time? or probably to land up in a very, very good college, whatever you're dreaming forward for. I hope it's the second one that I'm talking about. So let's be focused on our goals. Let's be all good children. Let's be all good friends, right? Ma'am, previous year questions exactly would not repeat, my dear children. The concept could be repeated. So that is why we say you should be having question practice. Okay. Chalo. Option. Yashmita has said 11B is the answer. Let me see. Shardhama says A. Azim Venkat says B, A, B, C, D. Chalo. Everyone is giving me options. So I hope now everyone is serious. Yes. So, live R strain, DNA, S strain. Okay, DNAs. Listen to me carefully. I'm going to make you hear a story. Okay, listen to me carefully. I have two strains. One is R strain, other one is S strain. S strain means it is smooth. Why is it smooth? Because, my dear children, this is a bacteria, pneumococcus bacteria. This is also a bacteria. This is different strains of the bacteria. Now, the smooth one is smooth because it has an extra layer around it. But you know what? Everything that looks good might not be good. So, which is why this guy is the actual culprit. If this guy goes and infects an organism, it will die. It will die. So now we have four scenarios. Listen to me carefully. First scenario, I am going to put live R strain. I am going to put live R strain into what? Into mice. And you know what? Mice lives. It does not die. It lives. Because R strain is not a problem. Now, that is rough strain, okay? Now, the second thing, I am going to put live S strain. I am going to put live S strain. Now, when I put that into the mice, my dear children, one sad thing happens. It dies. It dies. Okay. Now, going ahead, third situation, I am going to heat kill. I am going to treat this particular thing with heat. So, this is known as heat killed S strain. When I am going to put heat killed S strain, my dear children, into the mice, what will happen? Surprisingly, the mice will live. It won't die. It will what? Live. Because probably some changes is happening because of the heat in the S strain, which is why the mice is able to live. Now, the fourth thing is the most surprising thing. Okay, Griffith, he took live R strain, which initially was not causing any problem. He mixed it with heat killed, which was also not causing any problem, heat killed S strain. And then he put it in the mice, and then he found that the mice died. The mice what? Died, my dear children. Yes, the mice died. So when the mice died, my dear children, Griffith was like, oh my God, what happened? Initially, it was not happening like this. So then, he was not able to prove anything, but he said that probably something is getting passed from the heat-killed S strain to the R strain, transforming it. Transforming it to what? The virulent version, the S strain version, and killing the mice. Then Avery, McLeod, McCarthy came in the picture and tried out everything they could. So what did they do was, they took this same strain and they put RNAs. That means an enzyme that will cleave all the RNA. When they put that, still the mice was dying. Still the mice died. Then they put, instead of RNAs, they put protease. They put protein. Still the mice died, my dear children. Okay, there was no change happening, still the mice died. Then finally, they did something. They put DNAs. That means whatever DNA is going to be there, it is going to be cleaved. Okay, it is going to be cleaved. When they did this, my dear children, he found that the mice didn't die. The mice didn't die. Why? Because probably heat-killed S strain, my dear children, its DNA and everything got what? Cleaved. And it was not able to transfer it over here, which is why it was not able to convert this into S strain, and which is why the mice was safe. So he understood this by doing all this thing. So now, okay, now let's try to find the answer. So, the result of the following reaction carried out by Avery on Streptococcus pneumoniae has proven conclusively that DNA is the genetic material. By live R strain plus DNA from the S strain plus DNAs, did he use this to prove that? Yes, my dear children, he took the live R strain, he took the DNA from the S strain and he treated it with the DNAs. And now he found that the transformation was not taking place. Heat killed R strain plus DNA. Heat killed R strain. Why do you need to heat kill that? And DNA from S strain plus DNAs does not make any sense. Live R strain from DNA from S strain and RNAs, RNA cannot be cleaving the DNA out there, so it does not work. Again, transformation will still take place. Live R strain plus denatured DNA of S strain plus protease. Here also denatured, DNA is already denatured, so how will the transformation take place? So what is your answer? Your answer becomes A. Okay? Your answer becomes A. Children, can you put it down? I am not able to see. I didn't start to read what. Okay, Shanmuga Priya. Okay, you didn't start to read. Okay, you know that you didn't. So probably from now onwards, if you're properly done with your PU2, please start solving as many questions as you can from your PU2 board. Okay, it will be really, really helpful for you. Children, so A is the answer. So all of you got A, you got it correct. Now, why not it's D option? Because here you are already denatured the DNA. Okay, DNA is already gone. And then you're putting protease. He didn't do this. This was not his experiment. Avery did this. Okay, Avery didn't use denatured DNA. He used this. Normal things plus DNAs. Okay, I hope finally you understood it. Now, which of the following events would occur if the lac operon of E. coli when the growth medium has high concentration of lactose? In the growth medium, you have higher concentration of lactose. Then which of the following would occur? The structural gene failed to produce polycistronic mRNA. So, structural gene fails to produce polycistronic mRNA, my dear children, do you think so? As in, it won't be able to code. Z, Y, A would not be coded. That is what it says. If lactose is there, it is said lactose is there, then what is going to happen? So, this is wrong, because if lactose is there, my dear children, this lactose acts as an inducer, right? It goes and bites with what? The repressor. Okay, it goes and binds with the repressor, and now the repressor is not free to come and bind over here. Hence, transcription will happen normally. So, this is wrong. Here it says it's failed. So, wrong. Okay, Amrita, I am coming to you. Okay. And now, the repressor protein binds to RNA polymerase and prevents translation. No, my dear children, it won't be binding because the repressor protein is busy with what? Lactose. So, this is again wrong. The repressor protein attaches to the promoter sequence and represses the operator. Array, repressor is with what? Again, as I am saying, it is with inducer, that is lactose. The inducer molecule binds to the repressor protein, yes, and RNA polymerase binds to the promoter sequence. Listen, so there was an operator region, there was a promoter region. RNA polymerase was not able to move forward because this guy was coming and binding over here in the operator region. But now, since this guy is free, it's not binding, this is free, RNA polymerase will come and bind and will go ahead with transcription. So, this becomes your answer. Okay. So, all of you who said D is the answer, you are absolutely correct. Now, let me read out a question and you read out this. Okay. Amrita was asking, ma'am, I know the topic, but I don't understand the options. What to do? You have to invest, you have to practice more questions first of all. When you do the question practice again and again, you see in how many different ways they ask the question. So, I will suggest you go ahead and practice more and more. There is no other shortcut to it. Anyway, we have to understand the question, right? So, if you and also do not feel demotivated while seeing the question. You might be very much, what do you call it? You might be demotivated, which is why also you might feel that, okay, I am not understanding. So, have this motivation that, yes, I can do it. I'll be doing better. Okay? Chinmay, these questions are easy, da. And I have taken certain questions so that specifically when it comes on the day of examination, it becomes easy for you if you see it before. Okay, so Shashank, Shardhamma, Megha, Jilani, Azim, Vanita, all of you telling me the answer. Let's see. Out of 23 pairs of chromosome in male first and the 46th chromosome, that is the last chromosome. What is the 46th chromosome? Y chromosome, smallest chromosome, okay, respectively possesses how many genes? So, it is the smallest chromosome, so smallest number of genes also, right? So, your answer becomes the first chromosome has 2968 genes, and the last one, the Y chromosome, has 231. This is given in your NCERT. Okay, so what is the answer? C is your answer. Sania, Suvidha, Kalpana, Suma, all of you got it correct. Now do the 13th question. Do the 13th question. Read the following statement and choose the correct option. Nitrogenous base is linked to the pentose sugar through N-glycosidic link. My dear children, this is pentose sugar, okay, pentose sugar, 1, 2, 3, 4 and 5 carbon, that is why it is pentose sugar, right? Now, this is attached, the first carbon onto which nitrogenous base is attached. So, nitrogenous base is linked to the pentose sugar through N-glycosidic linkage. Is this true or false? It is absolutely true. This you call it as N-glycosidic linkage, true. Now, phosphate group is linked to 5-OH of a nucleotide. So, phosphate group is where, my dear children, on the 5th carbon. So, over here only it will come and attach. So, this is also correct. This is also correct. Now, going ahead, 2 nucleosides are linked through 3,5N glycosidic linkage. Do you think two nucleosides? Why do you need nucleosides? First of all, it should be nucleotide. So this is absolutely wrong. The whole structure that you see, my dear children, it is a nucleotide. Again, when you are binding it one more, you call this as nucleotide. This nucleotide, the next nucleotide. It's not side, it is nucleotide. Okay. Now, negatively charged DNA is wrapped around positively charged histone octamer to form nucleosomes. Yes. Okay. So what happens? DNA is very long. We have to fit it in the nucleus. So we have to coil it around the histone proteins. How many of them? 8 of them. Okay. So histone proteins, they are positively charged, and DNA is negatively charged. So that is why they come together. So that is also correct. Now, fifth one, chromatin that is more densely packed and stains dark is called euchromatin. Chromatin, my dear children. So over here, this particular unit is known as nucleosome, DNA wrapped around the octamer. Similarly, you will be having multiple units like this. Together, you form a bead on a string-like structure known as chromatin. Now, suppose they are close to each other, you call it densely packed. If they are loose, you call it loosely packed. Okay, the densely packed one, you call this as heterochromatin. You call this as heterochromatin. The loosely packed one, you call this as euchromatin. So over here, ulta is given. Here it is given chromatin that is more densely packed and stains dark is called euchromatin. Wrong. It is heterochromatin. Okay, and the chromatin that should be loosened, that will be euchromatin. Okay, which one is the transcriptionally active chromatin? Can you tell me? Which one is the transcriptionally active chromatin? Anyone in the comment section below, can you tell me? Alan, Mike, can you tell me, dear, what you want me to explain? This question, whole question? Euchromatin. Euchromatin is something that will be transcriptionally active. As in, for example, my dear children, if this is the histone octamer and the other one is over here, you have this much strand of DNA which is going like this. So basically, RNA polymerase can come over here, bind and do the transcription also. So that is why we call euchromatin as transcriptionally active chromatin. All of you who told this are correct. Okay, you got it correct. So what would be your answer for this question? 1 and then 2 and then 4 are correct. Rest everything is wrong, right? 1, 2 and 4 are correct. Wait. That means 3 and 5 are wrong. That means C is the option. Okay. Shobha Murthy class is boring, dear. Ayyo. Can't do anything. Fine, children. Going ahead with the next question. So I hope everyone understood. I'll just revise it once more. Listen to me carefully. Nitrogen base is linked to pentose sugar through N-glycosidic linkage. Yes, the sugar is there. Sugar binds to the nitrogen base through N-glycosidic linkage. Next question. Phosphate group is linked to the 5-OH of the nucleotide through phosphoester linkage. Yes, the phosphate group is actually bound over here in the 5th carbon. Okay, and this is known as phosphoester linkage. True. 2 nucleosides are linked to 3,5N glycosidic linkage. First of all, you need not link 2 nucleosides. Nucleosides means you remove the phosphate over here and you attach just a nitrogen base. This is known as nucleosides. You don't need to attach them. So it's wrong. Negatively charged DNA is wrapped around positively charged histone. True. Chromatin that is more densely packed and stains dark is called heterochromatin, not euchromatin. Okay. Going ahead. Question number 14. Which of the following radioisotopes is not suitable for DNA labeling based studies? Can you tell me the answer for this? Read the following statement and choose the correct option. Shanmugha Priya. Ma'am, in board I got 231 in PCM. How many marks should I? Children, you should not be asking this question. Aishwarya Ma'am has said not to answer the question, this type of questions. Because listen, you give your best, that should be your target. That's all. Okay. Chalo, which of the following radioisotopes is not suitable for DNA label studies? So someone says, okay, Chinmay, Preeti, Harshita, Sania, Kavya, Azim, Chetna, Suma, Batul, Moinili I explained, okay, and Sania, Tanushri, Kalpana, okay, all of you are attempting. Let's see who all are getting correct. Okay. So my dear children, if you're talking about DNA, what all does DNA have? If you want to radio label it, if you want to see it clearly, you should be tagging the things that makes up the DNA. What makes up the DNA? It's the sugar, it's a nitrogen base, it's the phosphate group. Obviously in the sugar you have hydrogen, so obviously you can target this. Okay, you have phosphate group, so obviously you can target this also. You have nitrogen base, so you can target this also. But do they have sulfur? Do they have sulfur? No. Which is why this is not suitable for DNA labeling experiments. So S35 is radioactive sulfur, and sulfur you mostly use for tagging proteins because proteins would be having sulfur in them. Okay, works? So which is why your answer is D. Chalo, going ahead with question number 15th. Okay, correct. Chinmay got it correct. So great. Ma'am, sorry for interrupting. Where can I get session PPT in WhatsApp channel? We would be putting down a link, then only we would be able to download it. So once after the class ends, I would be doing that. Okay, Yashash? Okay. Now compare the statement A and statement B. Chalo, let's become hyperactive children. Statement 1 says, a monocistronic mRNA can produce several types of polypeptide chain. What do you mean by monocistronic mRNA? This is a cistron. This cistron contains only one structural gene coding for one particular protein. Only one gene is there. Only one particular gene is there. One gene. This is monocistronic. So do you think a codon, basically a monocistronic mRNA, can produce several types of polypeptide chains? Can it produce multiple proteins? No. One gene, one protein. Okay, one gene, one protein, it will be producing it. So this would be wrong. Now, the terminator codon is present on the mRNA. The terminator codon, my dear children, is it present on the mRNA? Yes, towards the 3' end of the mRNA, you have the terminator codon. So is this statement correct? Yes, ma'am. Monocistronic mRNA, where do you find it? Eukaryotes or prokaryotes? Monocistronic mRNA, where do you find it? Eukaryotes or prokaryotes? Can you tell me that? And I will be telling the answer for this. So statement A is wrong and statement B is correct, which is why C becomes the correct option. All of you who got C as the correct option, you are doing great. Okay? Eukaryotes, exactly Harshita, exactly Sujata, Vanita, all of you are correct, my dear children. Okay, all of you are correct over here. So, your answer is basically monocistronic gene. Wherever you have a cistron carrying only one gene coding for one protein, you find it in eukaryotes. Okay, and the polycistronic, where one particular cistron have multiple genes, my dear children, coding for multiple proteins. Okay, it will be known as polycistronic, you find in prokaryotes. Works? Going ahead to the next question. 16th question. Can you try this question? E. coli cells with a mutated Z gene of the lac operon cannot grow in medium containing only lactose as a source of energy because what? Because of what? E. coli with a mutated Z gene. So of lac operon, my dear children. So you know in E. coli that is a prokaryote, and I just told you one structural gene has over here multiple genes. That is, there is Z gene, there is Y gene, and there is A gene right over here. So suppose the Z gene is mutated. So what will happen? Cannot grow in medium containing only lactose as a source of energy because of what? You are saying D is the answer. Let's see if D is the answer. In the presence of glucose, E. coli does not utilize lactose. Wrong. Lactose. They cannot transport lactose from the medium into the cell. Wrong. The lac operon is constitutively active in these cells. They cannot synthesize functional beta-galactosidase. That is the answer. My dear children, lactose, suppose it's there. There should be someone to break down this lactose so that the E. coli can use it. Who is that someone? It is beta-galactosidase. So Z gene codes for beta-galactosidase. So Z gene is mutated. Obviously, this is not produced. If this is not produced, can it break down lactose into glucose and galactose? The answer is no. So hence D becomes your answer. Okay, hence D becomes your answer. Kushal, can I explain it once this particular number of questions end? I will be explaining it. Okay. I will be Ajay, Y is for permease, exactly. So for this, I hope you got the answer. Everyone who got the answer, give me a thumbs up or a heart. Let's go ahead. Let's go ahead. Okay. Chalo. Now find the mismatch. RNA polymerase 1. Thank you children. We will be doing it. Okay. RNA polymerase 1 transcribes rRNA. RNA polymerase 2 transcribe precursor of mRNA, and RNA polymerase 3 transcribe tRNA, 5S rRNA and snRNAs. Do you think it's correct? Do you think it's correct? Which of them are correct? Can you tell me? So let me read out the names of the children. Rehu says A. Okay, Shashank says A. Chinmay says A. Suma A, Shashank, Pallavi Heart, thank you dear. Amrita A, Sania A, Shanmukha D, okay. Sania A, Preeti A, hi Nagaveni, Chetna, Lakshmi, Sujata, Jilani, Sachin, Aisha, Mira, oh my god, so many of you. Thank you, you are responding, that's so great. Now listen to me, ma'am, let's crack KCET. Yes, let's crack KCET. So RNA polymerase 1 definitely transcribes rRNA. Always remember, if you tend to forget it, RNA, ribosome is made up of RNA, my dear children. So the main priority of RNA polymerase is to make ribosomal RNA, okay, because both starts with R, you can remember like that. So RNA polymerase 1 first priority is rRNA. RNA polymerase 2 second priority is to transcribe basically DNA into hnRNA and then basically mRNA. So that is also correct. Now the last priority, third priority is basically to transcribe tRNA, okay, transfer RNA. So basically these three things are correct. So all of them are correct. All these are correct. So let's say ribosome contain protein. No, ribosome is made up of two things, rRNA and also proteins. Okay, rRNA and also proteins, ribosomal proteins. Works? So all of this is the correct answer. All of you got that. You are doing great. None of this are. Wait, that is wrong, okay. It would be fine. Okay, fun. Children. Find the mismatch. That is the question. I was correct over here. Find the mismatch. So they ask for the mismatch, which is by B becomes the answer. None of these. So listen, under pressure, even I who put the answers like this, I am also taking things in a different manner. So under pressure you might also do that. So which is why we should be both staying calm, right? Yes, we would be staying calm. So I hope you guys rectified. B is the answer, okay, because they asked for the mismatch. Now question number 18, histone octamer differs from nucleosome in lacking. In lacking they are asking. They are lacking something. Histone octamer, that means basically this is the histone, the protein, okay, and it differs from nucleosome. What is nucleosome? When there is histone octamer and also DNA. So what is the difference? I have already explained. Lysine and arginine. In both nucleosome and histone you find lysine and arginine. What are those? Amino acids, my dear children. Where do you find these amino acids? In the histone proteins. Histone proteins, the proteins are made up of amino acid, and both of them, they are highly positive, just like you and me. Just like you and me, they are highly positive, and the DNA wraps because DNA is negatively charged. Non-histones. Do you find non-histones? Histones octamer differ from nucleosome. Basically, neither nucleosome nor the histone proteins have non-histones. So non-histones are something else. Okay, there are other proteins other than histones. So both of them do not have it. So this is also wrong. Negatively charged DNA, yes. Only nucleosome have it. Okay, nucleosome have it. Histone does not have. Histone is just a protein. So this basically, can I say, is the answer? Can I say this is the answer? C becomes your answer. LKG, UKG, yeah. Is that easy? Is that easy? So, yeah. Question number 19th. In genetic coding, triplet codons make 64 possible codons. A doublet codon system would have made possible what? Ma'am, does this video contain 11th portion? No, the part 2 video will be containing 11th portion. This video contains all the important chapters. Okay? Children, try this. You were saying that it's an easy question, right? Now you try this. You'll say this is also easy, ma'am. Okay. Sujata, what do you want me to explain, dear? Can you repeat the question number? I'll explain. Ma'am, can you explain it again? 18th question once again. Okay, I can explain. Listen, histone octamer means this is histone, okay? Okay, I will do like this. When histone basically is wrapped by DNA, we call this as nucleosome. So this is histone and that is nucleosome. Both of them have lysine and arginine. Do they have? Yes, because histone is made up of lysine and arginine. Okay, so both of them have. You have to find what is different in them, right? Non-histones. Do they have non-histone proteins? Neither this has non-histone, other type of proteins. Neither this have other type of proteins. So this is also wrong. Negatively charged DNA. Histone does not have any DNA. When it is covered by DNA, you call this as nucleosome. So this is what is different between both of them. Histone octamer differs from nucleosome in lacking. What does it lack? It does not have the negatively charged DNA. I hope that is correct. Now you got it. Okay. All children who asked for explanation, I hope you got it. Now question number 19th. Human reproduction would be coming in, okay, in this part only. Okay, in genetic codon, triplet codons make 64 possible codons, my dear children. 64 possible codons. A doublet codon system would have been made possible when what happens? Basically, listen, how did we come to a conclusion that we should be having triplet codons, A, U, G and things like that? We have four, what do you call it as, nucleotides, my dear children. Four different type of nucleotides, DNTPs. One is A, the other one is T, G and C. Now we need to find different combinations. We need to find different combinations such that they produce at least 20 amino acids, such that they code for at least 20 amino acids. So four of them are there. If I do square of them, if they come in doublet pair, so 4 square, I'll get 16. I get 16 things, 16 unique things. Do you think 20 is more than 16? Yes, it is. So we need to make 20 amino acids, and for that these many codons are not enough. It's just 16 codons, right? So this is not true. Now going ahead, if it was like this 4 cube, then it would have been 64. We have 64 codons, and in 64 codons, my dear children, at least 61 of them codes for 20 different type of amino acids. You see repetition also. So this was something that was approved. Now then the question it is asked, if it was this case, doublet codon system, then how many different type of codons can you expect? Only 16 of them you can expect. So your answer becomes what? Your answer becomes what? D, my dear children. Answer D is final. So all of you saying A, B, C, D. For me, it's not A, B, C, D. I hope you got this answer now. It is only D. Okay, it is only D. Vaishnavi, very good evening, dear. Okay, children, these type of questions could come, so please be careful. Now next question, find the mismatch. Find the mismatch. So over here 174 bacteriophage has 5386 nucleotides. Bacteriophage lambda has 48,502 base pairs, okay, and E. coli has 4.6 into 10 to the power 6 base pairs, and haploid DNA is 6.6 into 10 to the power 9 base pairs. What is the answer? Everyone got it correct because this is so easy, right? Yes. So haploid human DNA, my dear children, it will contain 3.3 into 10 to the power 9 base pairs, half. This is of diploid. So in primary oocyte, tell me this, okay, in primary oocyte, what is the content of DNA? Is it 6.6 into 10 to the power 9 or is it 3.3 into 10 to the power 9? In primary oocyte, if the question comes in primary oocytes, what will be your answer? Chalo, fast. Ajay says 6.6. Yes, if primary oocyte, then it would be 6.6. Because primary oocyte, my dear children, it has not started dividing. It has just started off meiosis, but it has not divided into 2. So it will be having 6.6. If you are asked about secondary oocyte, it will be half because it has started meiosis. Okay? Okay? Break, Baba, I will give you later. Okay, we will have to finish little things. Now, for those of you, I think I have finished all the questions. No. Okay, question number 21. Question number 21. If the DNA of mammalian cell contains 6.6 into 10 to the power 9 base pair, the length of DNA would be, what is the length of the DNA, my dear children? What is the length of the DNA? If we have 6.6 into 10 to the power 9 base pairs, what would be the length of the DNA? So someone says B, someone says C. Okay, children, if this is a DNA, in between each base pairs there is 0.34 nanometer gap. There is 0.34 nanometer gap. When you convert it into meters and all, when you convert into meter, it's like 10 to the power 9 minus 9. Okay, when you convert into meter. So basically, if you do into multiplication, so 6.6 into 10 to the power 9 base pairs multiplied by 0.34, you will get around 2.2 meters. Okay, you will get 2.2 meters. So this becomes your answer. C becomes your answer. So all of you got C. Kudos to all of you. I'm so sad I could not read out your name because it's going like this. I'm so sorry, but all of you are giving the answers. God bless you all. You're doing an amazing job out there. Okay, you're supporting a teacher right there. So yes, Now DNA differs from RNA in having what? How does DNA and RNA differ, my dear children? 22. Oh my god, Preeti is so fast. Chetna fast. Okay. Chinmay. Okay. Okay, D. Oh my god, so many people are saying D. All of this. So deoxyribose sugar instead of ribose. Yes, in the case of RNA, you have ribose sugar. In the case of DNA, you have deoxyribose sugar. So this is correct. Thymine instead of uracil. Yes. Okay, thymine is there in the case of DNA. In the case of uracil, it is RNA. In the case of uracil, it is found in RNA. Now, H instead of OH at the second carbon position. So this is a sugar, pentose sugar. So this is first carbon, this is second carbon. Over here, there will be OH in the case of ribose sugar, and that is in the case of RNA, and there will be just H in the case of DNA. So H instead of OH, that is also correct. So I'll say all of them are correct. D becomes your correct answer. Okay? Children, great. Now, listen to me. For those of you who have completed this chapter, and now those of you who were asking lacopron from me, listen carefully, okay? I'll just take 2 minutes of your time. Listen to me carefully, you will never forget. Okay, lacopron story. So, what do we have? Over here, we have 2 structural genes. One structural gene over here. For each structural gene, you have a promoter region. This also will be having a promoter region. What is this gene coding for? This is coding for a regulatory protein. A regulatory protein. That means something that will regulate negatively or positively. Now, over here, you have an operator region and you have 3 genes. 1, 2, 3. Z, Y, A. Z codes for beta galactosidase. Okay. Y codes for permease. And A codes for trans-acetylase. So, the condition is when you have, this is happening in E. coli. This is happening in E. coli. Now, E. coli, when it gets lactose, it becomes so happy. It becomes so happy. And it will break down lactose with the help of beta galactosidase enzyme into what? Into glucose plus galactose. Okay. Permease enzyme is there for basically increasing the permeability of the membrane so that the lactose can get inside. Trans-acetylase deals with everything that is formed in between, all the metabolites that is there. Now, this is the usual scenario. But what happens is that mostly this this regulatory protein codes for a repressor protein. This regulatory gene codes for a repressor protein, and this repressor protein says, I would not let this happen, I'll go and bind on the operator region. It goes and binds on the operator region, and what will happen? The RNA polymerase, which was supposed to transcribe the whole thing, is not able to move ahead because there's a big repressor protein right there. Okay, this is the usual scenario, and you say basically that this is switched off condition. This is what? Switched off condition. Now, when lactose is provided in the medium, my dear children, what will happen? Suppose lactose is there in the medium, lactose got inside. Lactose binds with this repressor protein, okay, and hence the repressor protein forgets to bind over here. Now what is happening? RNA polymerase is free. It lets this whole thing undergo transcription. So lactose, can I say it is an inducer? Because it is inducing what? Switch on condition. So it will get switched on and it will do all the stuff needed. I hope that is clear. Okay, explain fast, ma'am. Explain fast, ma'am. Oh my god. Children, those of you asked for the lacopron, I hope that's clear and you will never forget it. Please, you have to remember it. Okay. Now going ahead to the next chapter, that is sexual reproduction in flowering plants. Okay, Kushal K. Mathod, you're most welcome, dear. First question. Arrange the given statement in correct order of their occurrence. Male gametes enter the cytoplasm of the synergids. Generative cell divides and produces 2 male gametes. Pollen grain possesses generative cell and tube cell. On breakage of synergid, 1 male gamete fertilizes with the egg cell. Tell me the answer fast. You are telling me to do fast, now I'll tell you to do fast. Children, let me breathe also. Sanvi, thank you so much, dear. 23B, okay. Preeti is back, Chetna is back, Vishwa is back, okay. 23B you are saying. My dear children, first thing, you're talking about pollen grains. So yes, pollen grain has a generative cell, bigger cell, and, there is a vegetative cell, and there is what? A generative cell and a generative cell. This generative cell divides into 2 and you have got 2 male gametes. what happens to the nucleus of the vegetative cell? It becomes the tube nucleus. It becomes the tube nucleus. It will be helping in the formation of pollen tube. Fine. So pollen grain possesses a generative cell and a tube cell. First one, correct. Okay. Now what happens? The generative cells divides and produces 2 male gametes. That is the second thing. Yes, ma'am. Correct. Now what happens? The male gamete enter the cytoplasm of the synergids. My dear children, this is embryo sac. You have 2 synergids and then the egg. So basically, pollen tube, when it enters, it has to break open the synergid, and the male gamete, then only it can get entry. So this is the third thing. And this one, on breakage of synergid, 1 male gamete fertilizes with the egg cell. 1 male gamete will fuse with the egg cell. So this is the fourth thing. So 3, 2, 1 and 4. 3, 2, 1 and 4, B becomes your answer. Okay? Works? Next, 24th question. After how many divisions does the functional megaspore result into mature embryo sac? Can you tell? Children, cool down. It's okay, it's okay. So all of you who are going to say bad comments or something disturbing the class, I have the whole right to block them, right? So do not worry, I'll handle them. But thank you for standing up. Yes. So after how many divisions does the functional megaspore result in mature embryo sac? Have you got any idea? So someone says D, Chetna says D, Preeti says B, Vishwa says C. So I've got all the A, B, C, D option. My dear children, you have one functional megaspore, right? And this functional megaspore, it divides first, there is first mitosis, where it divides into 2 nucleus. Okay, then again mitosis, where it divides into, this divides into 2 and this also divides into 2. So now we have 4. Again mitosis happens, the third one. So this is the first one where it divided into 2 and then into 4. And now the third one, basically this will divide into 2, this will divide into 2, this will divide into 2, this will also divide into 2. So 1, 2, 3, 4, 5, 6, 7, 8. At the end we get 8 nucleus. So almost how many divisions are there? After 3 mitotic divisions. So this becomes your answer. Okay, B becomes your answer. Children, is it fine if we give break after this? After sexual reproduction flowering plants, we will have a break, okay? All of you, I hope you will never forget it as well. So 3 mitotic divisions are there to form a mature embryo sac. Now, going ahead, in Camelina, flowers are produced on underground rhizomes or root, aerial branches. Aerial branches means at the top you have at the top air branches. And then for example, this is the land, these are the aerial branches. So underground rhizomes or root, aerial branches, leaves, or both A and B. What do you suggest? What do you suggest? D. So someone says D, all of you are saying D, or someone says A also. Yes, your answer is actually D. Camelina, my dear children, it has 2 types of flowers, if you know. Camelina, viola, or oxalis, all of them are clistogamous as well as chasmogamous. They have both the flowers. So the clistogamous flowers are over here and they would be closed, my dear children. And the chasmogamous flowers, they will be they will be open like this. So they have both. So your answer is D. Now in your NCERT, the picture is given. Even I didn't believe it until I saw the picture. So please see the picture once. Okay? Ma'am, D. Yes. D is the correct option. Now question number 26. Study the following statements and choose the correct option. Okay? Are we ready? Chalo. Tapetum nourishes the developing pollen grains. Does tapetum nourishes the developing pollen grains? Yes, it does. So you have multiple layers. The outermost layer is epidermis, then you have endothecium, then you have middle layer, and the innermost layer is tapetum, which nourishes the sporogenous tissue, which has the ability to form the pollen grains. Okay, so this is correct. Now hilum represents the junction between ovule and funicle. Does it? Does it? My dear children, over here, this is the ovary. Inside that you have the ovule. This will be placenta. Placenta attaches to the ovule through the funicle. This is what? Funicle. And the attachment point is known as hilum. So hilum represents the junction between ovule and the funicle. Correct. Now in aquatic plants such as water hyacinth and water lily, pollination is by water. You know, that's a crazy fact. Both of them has the name water in it, but they do not get pollinated by water. This is wrong statement. This is what? Wrong statement right over there. Now the primary endosperm nucleus is triploid. How do you get the primary endosperm nucleus? When there are two polar nuclei, the two polar nuclei fuses with the male gamete, second male gamete. Okay, so over here two polar nuclei is haploid, haploid, and this is also haploid. N plus N plus N is what? Triploid. So basically primary endosperm nucleus is triploid. That is also correct. So can I say 1, 2, 4 are correct? 1, 2, 4 are correct. That means all of you said B is correct. B is correct. Okay, B is absolutely correct. Chalo. Yes, Vishwa, they are going to show wind pollination, not water pollination. So yes. So great. Next question. The male gametes of rice plants, okay, let's see who all get this correct. The male gametes of rice plants have 12 chromosome. Male gamete, okay, gamete has 12 chromosome. Now, the chromosome number in the female gamete. The gamete will always have 12, my dear children. Half, okay, the gametes will always have half because these gametes have to come together. The male gamete and the female gamete has to come together to form the zygote, right? So basically, the female gamete is also 12N, okay, 12. Now zygote is basically formed when these two guys fuses, so it will be 24. Now the cells of the seedling as in the zygote, it will continue its division and form a whole new seed out there, right? So it will again be 24. So 12, 24 and 24 should be the thing. So you are saying C, so I will also say C. I'm not even checking, it's C. Okay, works. All of you are doing absolutely a great job answering all the questions out there. Awesome, children. Okay, crazy, it's fine. There is no trouble happening, my dear child. It's all cool. Okay, now the 2000-year-old seed excavated from the King Herod's place at the Dead Sea belongs to what? Okay, Preeti says it's B, Yashwita says it's C, Preeti B, C. I see so many things. Ma'am, I'm getting wrong answers. Harshita, it's fine. You learn from this. Okay, it's fine. You know what, in exam-like condition, there won't be 10,000 people answering around and it won't be like this. Okay, it will be easy for you. Trust me. I'm thinking C, but typing A with full confidence. It happens. It happens, my child. So your answer, all of you who said Phoenix dactylifera, you are absolutely correct. So Phoenix dactylifera you found in King Herod's place, okay, that is 2000 years ago. If the question was 10,000 years old viable seed, if the question was 10,000 year old viable seed, where do you find that? Where do you find that? It would be Lupinus articus. It will be Lupinus articus if you're talking about 10,000 years old viable seed. Okay? Cool? Yes, Bhumika. Yes, Chetna. All of you children. Great. Now, okay, try this. 100 functional megaspore gives rise to how many embryo sac? 100 embryo sac, 50 embryo sac, 25 embryo sac, 200 embryo sac. C, C, A, B, C, D. Chalo. Arctic tundra, yes, Lupinus arcticus is Arctic tundra. So say some people are saying A, so I will go with A. Next question. So functional megaspore. Listen, 4 megaspores are produced, but 3 of them degenerate. Only 1 is functional, and this functional megaspore, my dear children, will be giving rise to 1 female gametophyte, that is embryo sac. 1 embryo sac. So 1 functional megaspore, 1 embryo sac. So 100 of them, that means 100 embryo sac, and this becomes your answer. So who all got A as correct option? Kudos to all of you. Now 30th question. Autogamy can occur in chasmogamous flowers if autogamy. What do you mean by autogamy? That means there is a flower and here you have the female and the male part. Now you want this to happen. The pollen grain should be landing over here and the stigma. Okay. Then my dear children, what should be answer? Autogamy can work in chasmogamous, the open flowers. Usually open flowers are there, so other pollen grains also can come and do things like that. But it will work. C, C, okay, wait. You are saying C, D, B, C, okay, C, C, C. Pollen matures before maturity of ovule. Do you think that could happen? If it happens, then obviously it cannot come and fertilize, okay? Ovules mature before maturity of pollen. Again, there is no synchronization. Both pollen and ovules mature simultaneously. There is synchronization and hence this becomes the answer. Anther and stigma are of equal length wing. I know, I understand that it is correct, but just understand one thing, okay? Suppose they are equal length wing, but the synchronization is not happening. They are not maturing at the same time. Do you think autogamy can happen? No. So what is more important? Both pollen and the ovules mature simultaneously. This becomes the most appropriate answer. So C is the answer. This is why I gave the question as well, because I know D and C is quite confusing. Okay? Ma'am, ಸ್ವಲ್ಪ tough questions give. You know what? KCET is not about tough questions. It's about asking from basics, and you do not understand the basic. That is the problem. Okay? Chalo. Next. In the embryos, question number 31. In the embryos of a typical what? I think, okay, typical monocot plant, the structure enclosing leaf primordia and a single cotyledon are respectively what? Can you tell? Crazy, I cannot see Alex over here, so I cannot block him as well. I think, I'm so sorry, I'm not able to see. Okay. Children, what do you call this? Tell me the answer. So there is something that encloses the leaf primordia. So you might have studied, there's an embryonal axis like this. From here comes a cotyledon that is known as scutellum. What is? It is known as scutellum. Monocots has this one cotyledon that is known as scutellum. Okay. Now, over here, you have this region which is known as plumule, giving rise to the shoot. This is known as radicle, which gives rise to the shoot. Sorry, root. Okay. Now, there is some region like this which covers the leaf primordia, and this is known as coleoptile. This is known as coleoptile. Okay, and the cotyledon, the only cotyledon is scutellum, which is why, just a second. Okay, which is why your answer would become what? Coleoptile and scutellum. B. Cool? Works? So all of you who got B as the correct answer, great. Now, question number 32, you have to find the mismatch. False fruit other than ovary, thalamus taking part in fruit formation. Is it correct? Begum, tomorrow her live will be there. Okay? Now, false fruit other than ovary, thalamus taking part in fruit formation. So, usually what happens, my dear children, you have the ovary, then the ovule, right? So, ovary, it is the one that becomes the fruit. But in the case of false fruit, apart from the ovary, other things can also help in the formation of fruit. So, this is correct. Okay, you have to find the mismatch. Parthenocarpic fruits developed without fertilization. Can it happen? Yes, that can also happen. When does that happen? When they have certain hormones such as auxin and things like that. At this point of time, there is no need of fertilization. The ovary will become the fruit without fertilization. This is also correct. True fruit developing from the ovary. This is also correct. Seeds developed from the ovary. Wrong. Seed basically develops from the ovule. This is the seed and this is the fruit. Fruit develops from the ovary. So your answer is D. Okay, 32D. And with that, we are going ahead to the next chapter. Any doubts that you have in this particular chapter or any concept you want me to explain in sexual reproduction in flowering plants, I can explain. Listen, you have to make sure that you are going through. Okay, tell me one question. I will be randomly asking questions. You have to clear them up. Okay? Okay, are you ready? Are you ready? I'll put it on a blank page. The first question that I am having is, okay, tapetum which is there. Does it have one nucleus per cell? Tapetum which is there, which provides nourishment to the sponogenous tissue, does it have one nucleus? Tell me the answer for this. Tapetum. No, yes, it has not one, it is binucleate condition. There is more than one nucleus because, my dear children, it does not undergo cytokinesis. Karyokinesis is done. The nucleus divide, but the cell does not divide. Okay. Now the next question. From one microspore mother cell, how many microspores or pollen grains do you get? How many pollen grains can be produced? For example, if I say 100 microspore mother cell are there, how many pollen grains will be produced over here? Can you try that? If 100 are there, how many pollen grains will be produced? 100 are there. Each of them, my dear children, divides in over here. MMC divides, undergoes meiosis, again undergoes meiosis. At the end, you get 4 of them. 4 4 microspores, which all of them give rise to pollen grains. So if 100 of them are there, so 100 into 4 gives 400. Remember that. Now if the question is, okay, if the same question is asked in a different manner, for example, if it's not microspore but megaspore mentioned over here, then what is your answer? Megaspore. If 100 of them are there, how many embryo sac is going to be produced? If 100 megaspore mother cell is there, then how many embryo sac is going to be produced? 100? 100? Yes. So why? 1 divides into 2, this also divides into 2. This is how meiosis happens. At the end you get 4, but only 1 remains functional, rest all of them degenerates. This one functional one is going to give rise to the embryo sac. So if you have taken 100 of them, you just get 100. There is no tripling, there is no doubling, nothing. Okay? There is only one functional megaspore which divides further or which forms the embryo sac. Okay? Works? Cool? Remember that. Now what? Okay, now in the case of in the case of embryo sac, tell me the ploidy of central cell. Tell me the ploidy of central cell. Central cell. So these two are synergids. This is the egg cell. Okay, these two are synergids. This is the egg cell. So egg cell. These are the antipodals. Over here you have 2 polar nuclei. I am asking you about the central cell, this big cell. What is the ploidy of that? You are saying diploid, everyone is saying diploid, and you are absolutely correct, my dear children. So all of them are having haploid. Only this central cell, because it has 2 polar nuclei, and each nuclei has N and N, which is why it becomes diploid. PEN, exactly, primary endosperm nucleus. Now, does the endosperm form first or the zygote form or the embryo forms first? Does the endosperm form first or the embryo form first? I am explaining the concepts because you will get questions from these. Okay? Endosperm forms first, right? Because endosperm has to provide nourishment. Now, my dear children, give me 2 examples. Give me 2 examples where perisperms provide nourishment. Where perisperm is there to provide nourishment to the growing embryo. Give me 2 examples of perisperm. What is perisperm, my dear children? Over here. Yes, black pepper and beet. So if you see, this is the ovule, yeah, and these cells are the cells of the nucleus, and one such cell is the megaspore mother cell which undergoes division, and usually this nucleus goes off, but sometimes it may persist and provide nourishment to this embryo which is going to be formed over here. So this is known as perisperm. So you find it in B square, that is black pepper and beet. Okay. Now a polyembryony, where do you find it? Give one example of polyembryony. Give one example of polyembryony. What do you mean by polyembryony? Multiple embryos are being formed inside the ovule. Where do you find it? Orange, yes, citrus fruits. You will find this in citrus fruits. Always remember. Okay, remember citrus, lemon and things like that for polyembryony. Works? Now, hear me out. Hear the statement correctly. Okay, I am telling a statement. Hear me out carefully. Tell me if it's true or false. Apomixis is the production of fruit without fertilization. Apomixis is the production of fruit without fertilization. Apomixis is production of fruit without fertilization. False or true? Why false? Tell me why it is false. Then what is it? Production of what? Production of apomixis is the production of seed, my dear children. It is producing seed. Parthenocarpy is production of fruit without fertilization. That is found in banana and things like that, where you do not have seed. Okay, because fertilization has not happened. Apomixis is the production of seed without fertilization. Always remember that. Okay? Yes, Harshita. See, you are getting the answers correct. Super proud of you, all of you who are getting the answers correct, my dear children. Okay? I hope that sums up everything. Do we have anything else? No. Okay, that's all. Cool. Let's go ahead with the next chapter, human reproduction. Malan Gowda, my dear. Okay, monocot and dicot. Listen, I will be just talking about one more thing and then we'll go ahead. Okay, just one more thing, listen to me carefully, okay. That is what monocot seed and the dicot, the embryo. Listen to me. So usually dicot embryo, it has an embryonal axis and two cotyledons. So they have two cotyledons, okay. This embryonal axis, it will end up in plumule and this will end up in radicle. The radicle will be having root cap. The radicle will be having root cap. Now we talk about monocot, they have an embryonal axis, but instead of two cotyledons, monocot, they just have one cotyledon and this is known as scutellum. Okay, here they have plumule and here they have radicle. Here also they have root cap. Here also they have root cap, and the root cap basically is surrounded by, coleorhiza, okay. R for root and R for coleorhiza, remember like that. Over here it is surrounded by coleoptile. So P for plumule and P here in coleoptile is also there. Apart from this, they also have persisting endosperm. Okay, that is why they just have one cotyledon. Fine. I'll give 5 minutes break. Okay, I'll give 5 minutes break. So this type of seed is known as albuminous seeds, which has the endosperm. This type of seed is known as non-albuminous seed. Fine. So your monocot example is something that you cannot break into two. For example, maize, rice, all this thing you cannot break into two. But here peas, okay, chickpea, you can break into two. So dicots are the example for that. Now over here, oh, you saw the answer, right? So we will take a 5-minute break, kutti break. 5-minute break, you can take the break and you'll be right back. And this time when you're coming back, it's 75 people out there, it should be 200 people. Can we make it 200? Share it with your friends who might need their help, okay? And let's cover all the other chapters. Just 5 minutes, I will also drink water and come. Can we make it 200? Let's make it 200 the target. Children, do not forget to drink water. I'll take a 5-minute break. Please take a 5-minute break. Your time starts now. It is 5:39 now. I take it 5:40, okay? Be back at 5:45. Be back at 5:45. You'll be like, ma'am does not give break as well. She just came before before time. So I will be waiting until 5:45 since that's the time that we set. But if you children have any doubts, you can ask me, okay? And I am very, happy with Chinmay, who said that let's ask questions to each other. Yeah, so maximum utilization of time. That is awesome. How much? Okay. Chirag, can you put it down in the comment section? I would be answering it to you there after checking, okay? We'll wait for just 1 minute, let all your friends come together, and as I said, let's make it 200 this time, okay? The maximum people who are going to watch this should be 200. Let's maximum people utilize it. Sania, ask questions. I will be doing it after human reproduction, okay? There's only 1 minute left, no? So let's have that zigger. All of you confident for cracking KCET 2025? So it's 5:54. Ma'am, what do you think this time KCET will be easy? I'll tell you something very honestly. KCET examination is generally easy compared to all the other examination. Okay, so I suppose it will be easy. But you have to know the concept. If your NCERT is full clear, it becomes all the more easy. Chalo, 5:45, let's start. Human reproduction, I'm going to start. All of you can ask your friends to join. Let's make it 200, getting things started. The correct chronological order of the following events. You have to talk about the events, okay? So let's start. Gametogenesis, fertilization, insemination, gestation, implantation, parturition. What is the order? Correct order, my dear children? First of all, gametes have to form. You call this gametogenesis. After that, gamete has to be transferred. You call this insemination. After that, both of them have to come together. You call this as fertilization. After that, basically this particular zygote will divide into embryo and later get implanted. This is known as implantation. And then the fetus will be growing. This is known as gestation. And last but not the least, the baby will be moving out. It is known as parturition. So your answer becomes C. All of you. Great. Okay, all of you doing great. Your answer is C. Who all got it correct are doing absolutely wonderful. Now, read the following statement about seminal plasma and choose the correct statement from the given options. Seminal plasma is secreted by seminal vesicles, prostate and bulbourethral gland. Second thing, it is rich in sucrose and calcium. Third thing, it contains certain enzymes also. So, what do you think is the correct option for this? What do you think is the correct option for this? I see 90 people have joined. Come on, let's make it 200. Ma'am, will this question help in NEET? Yes, it will. The difficulty level of NEET is much more higher, but I see the concept. If you make it strong, it will be good for you. So there are so many with D options. Yashwita, Deepankar, all of you saying D. Okay. D is what? All of this. You are absolutely correct. My dear children, seminal plasma is the secretion of 3 different glands. Seminal vesicle, prostate and bulbourethral. So usually children get confused, seminal vesicle, then it is seminal plasma. No, it's for all of them secretions together. Now it is rich in fructose. Why? Because the fluid that seminal vesicle releases, it has enough amount of, it is rich in sucrose and calcium. Children, over here, is D the correct answer? Can you think once more? Now if you think, is D the correct answer? It should be C. Why? Because over here, it is written sucrose. It is not rich in sucrose, it is rich in fructose. It is rich in fructose. So seminal vesicle has released a fluid that is rich in fructose, which gives energy to the sperm, my dear children. Okay, and apart from that, it is calcium which is there. You get it from prostate gland. Okay, prostate gland gives 2 things. One is calcium and the other thing is the enzymes. Okay, 2 things, enzymes and the calcium. And fructose by seminal vesicle. Bulbourethral gland helps in lubrication. So here sucrose is given, which is why this is wrong. So in that case, 1 and 3rd statement is correct. In that case, C is the correct answer. I think I have also marked D, but do not go with that. It's C what is the correct answer. So as of now, I saw sucrose. So this is the correct answer. Okay, got it? Not calcium is there. Okay, calcium is produced by the prostate gland. cool. Let's go ahead. Now, the ploidy of spermatogonia, primary spermatocyte, secondary spermatocyte and spermatid is what? My heart dropped for a moment when you said it's D. It happens. I like to do that. Okay. Now, over here, the ploidy of spermatogonia, primary spermatocyte. My dear children, again try to do. Please answer in 35 A, B, C, D. I won't understand which question you are answering, okay? You all are saying D. How you guys are so fast? Let me try it out. So, spermatogonia, my dear children, it's a normal cell, so it will be diploid to N. Okay. Now, primary spermatocyte, again, it is differentiated into primary spermatocytes, which is also 2N. Okay. Now, we are talking about secondary spermatocyte. So, that you get when you divide it, it undergoes meiosis 1. You get 2 of them. These are secondary spermatocyte, and it will be half. It will be haploid, so it is N. Now, spermatid, when this guy divides into 2 again, you get spermatid. This time there is no reduction. In meiosis 2, second round of meiosis, there is no reduction. It will be entirely same. It will be haploid only. So, can I say it is 2N, 2N, then N and N? Yes. So, all of you said D. You are absolutely correct. Okay. And it's crazy how speedy you are replying. Okay. It's awesome, awesome, awesome. Chalo, next question. The first meiotic division during oogenesis is completed at the stage of what? The first meiotic division. So, I hope you know that, my dear children. You have this thing, what do you call it as? Primary oocyte, yeah. Primary oocyte enters the stage of meiosis, but it gets stuck. It gets stuck. When does it complete its division to form 2 cells? One cell is polar body, first polar body, and the second cell that you have is the secondary oocyte. When does this happen? When does this division get completed? That is the question. So, someone says D, someone says B, someone says C. Let me see the answer. Primary oocyte within the primary follicle, primary oocyte within secondary follicle, primary oocyte within tertiary follicle. The answer is primary oocyte within the tertiary follicle. In the tertiary follicle has 3 things, okay? Not 3 things exactly. You have granular cell layers, you have granular cell layers, and then one, one antrum, there is inner theca interna and there is outer theca externa. This whole thing is known as tertiary follicle, and inside that we have a chunnu primary oocyte which is dividing into what? Secondary oocyte. So, why not D, ma'am? Secondary oocyte within the tertiary follicle. First meiotic division. At the end of it, what do you get? Secondary oocyte you are getting, right? You are asking the stage. So, this first meiotic division during oogenesis is completed at the stage of primary oocytes within tertiary follicle. Now, you have got me confused. Wait. Let me think, okay? Is completed at the stage of. Let's keep this question on target. Okay, we'll come back to this question. Okay, we'll come back to this question. Let's keep this question on target. Let's keep it aside. It is 36. My brain is not braining now. First meiotic division during oogenesis is completed at the stage of complete. Okay, we'll come back to it. Okay, now I have marked that, but yes. 37th question. Can you try this? Which set of hormones are secreted during pregnancy only? Yeah, try this. Which set of hormones are secreted during pregnancy only? Estrogen, HPL, relaxin, progesterone, cortisol, HCG, HCG, HPL, relaxin, cortisol, progestrogens, relaxin. What is the answer for this? This is easy. Your answer would be C. Your answer would be what? C. Why C? Because estrogen is released normally also in the case of females, yeah, without pregnancy also. Even progesterone to maintain the endometrium. So that is not something that is released only during pregnancy. Cortisol and progesterone, again, it can be released during normal time also. So this is something that is released only during pregnancy because, my dear children, HCG and HPL is produced by placenta. Placenta is formed only during pregnancy, and relaxin is produced by the ovary during later phase of pregnancy, but only during pregnancy itself. So C becomes your answer. Okay, works? Chalo. Next question. Embryo at 8 to 16 cell stage is called as blastula, morula, trophoblast, all of this. What do you call this? 38B. Shushmita says 38B. Children, put it number and tell me. Embryo at 8 to 16 cell is known as blastula, morula, trophoblast, or all of this. Your answer is morula. Suresh, what happened? You said disturbance is there. What kind of disturbance? It is known as morula, okay? 8 to 16 cell is morula, and then later on morula will keep on dividing from blastocyst, okay? From blastocyst. In blastocyst you have outer cell which is known as trophoblast and inner cell mass. This different thing. So your answer is B, morula. Going ahead. The fetal ejection reflex in humans triggers the release of what? The the fetal ejection reflex, as in the baby will try to move out. When it moves out, what happens? Basically the stress receptors on the cervix, they become activated and they will send signals to the maternal pituitary to release what? To release oxytocin. To release oxytocin. Now what will happen? This oxytocin will be coming towards the uterus, and in the uterus there is myometrium layer, the muscular layer, and on this, my dear children, you would be having certain receptors to receive the oxytocin. When they come together, they will make contraction of the muscles and the baby would be moving out. So your answer becomes what? Oxytocin from maternal pituitary. Oxytocin from fetal pituitary? No, baba. Okay, human chorionic gonadotropin from placenta? Wrong. Human placental lactogen from placenta? Wrong. Okay, so listen to me carefully. I am telling you two things. You have to listen to me carefully. The baby which is there, it tries to move out. This is a reflex. Okay, this is a part of fetal ejection reflex, my dear children, when it tries to move out, activates the stress receptors. Apart from that, even the placenta is also going to release certain thing. Even the placenta releases estrogen in very high amount, and these estrogen makes the receptors over there active, such that when oxytocin comes, it can bind over here. So two things happen. One is the baby moving out, and also the placenta releasing estrogen, and later the oxytocin comes into play. Okay, so yes, so your answer becomes D. Who is this one who is saying A is correct? A is not correct. Oxytocin from fetal pituitary? Wrong. Oxytocin from maternal pituitary, and pituitary gland, my dear children, it has two lobes, anterior and posterior. Oxytocin is released from the posterior lobe. Remember that also. Okay, KCET dress code, my dear children, we would be making a separate video for that. Okay. Disturbance in my voice. Everyone feels that? Suresh, can you please check it out once, my dear children? Children. Okay, chalo. In which period during embryonic development heart starts beating approximately 60 times per minute? First week, fourth week, second week, third week? Ma'am, there's a beep sound. All of you can hear the beep sound? Okay, so I see a mix of A, B and okay, all the things out there. So all of us hearing the beep sound. Oh my god. So there has to be a problem. How to fix that? Tell me if it's fine. That AC sound. Okay, then it's fine. Listen, you're talking about heart. So heart's complete development, basically heart starts beating, that's what you say, after 1 month. And 1 month is made up of how many weeks? It has even 4 weeks. So can I say at the end of 4th week you will be having heartbeat? Can I say that? Can I say that? Yes, after first month. After first month you have heartbeat. Listen to me carefully, this is important. Now what happens in the second month? Second month you will be having limbs and digits. You will be having limbs at the end of second month, limbs and digits. And also organ system is formed, and apart from that external genital organs are also formed. External genital organs are also formed in the second month. Okay, now what happens in the third month? Third month is skipped. Let's skip third month, let's skip 4th month, okay, because it's not there in your NCERT. Let's come directly to the 5th month. In the 5th month, what happens, my dear children? For those of you who were there with me in the previous session, I did all the action and I told you. Okay, I did all the action and I told you. In the case of, voice is very low. In the 5th month, I hope you're able to hear. Yeah, in the 5th month, baby says that I want to show off. Okay, so basically give me some hair, God. So God gives hair on the head, and also it is able to move around. So first movement, and also hair on the head. Hair on the head is found on the 5th month. Now what happens on the 6th month? Any guesses? 6th month, that is the last month given in your NCERT. God says that I gave you hair on your head. Now listen to me. You should be having hair on your body. So body hair develops in the 6th month. And apart from that, my dear children, your eyelids separate, your eyelashes also grown. So you have eyelid separate, and formation of what? Eyelashes as well. So will you forget this now? Suresh, children, okay. Everyone who says that I am not audible, can you say, ma'am, not audible? All of you together, so that I can see. Once more, through you, Harshita, you want to explain? I will explain you. Don't worry, okay? I'll explain right away. No, it's fine. It's fine. Everyone says it's fine. Someone asked me about cortisol. Cortisol, dear, it's there for blood sugar control, also for blood pressure control, the ionic control, electrolyte control, and all these things. That is why you have. Suresh, please check. Okay? No, it's audible. Chalo. Now listen to me carefully. Those of you didn't listen. Okay? I am going to do foolproof action and make you remember. First month, heart. Every time you die, what is the first thing that you check? When someone is about to die, or basically when we want to see whether they have died or not, we have to check the pulse, right? So basically you're talking about heartbeat. So the first thing when you're born, also the first sign of life is what? Heart. So heartbeat after first month. Second month, you have got limbs, you have got digits, you have got plenty of organ system and the external genital organ. Okay. Now fifth month, the baby says, I want hair on my head and I also want to move around. Okay. So hair on the head and movement is in there in the fifth month. Now sixth month, the God says that, hey, you should be having hair on the body. So body hair, eyelids are separated and eyelashes are also formed. Okay, which is why your answer becomes B. I hope that is clear. Cool. Chalo. Cortisol has multiple functions. Okay, it's not just one function, multiple functions. There are different types of it. Okay. Your explanation is superb. That is, thank you so much. Someone, okay, wait, okay, just give me a second. I'm trying to call up someone who can help me with this, okay? So just bear for a second. There's no problem. Okay, children, let's go ahead with the 41st question then. Until then, it will be coming back, okay? So, 50 secondary oocyte in female and 50 secondary spermatocyte in male gives rise to what? How many ova and how many sperm? So, 50 secondary oocyte, my dear children, if you know, we have to make oogenesis and spermatogenesis. So, all of you saying maybe C, A, B, C, D. So, let's see what is the answer for this, okay? So, over here, you have primary oocyte. We have to start with primary oocyte and we will start with primary spermatocyte over here, and with this you will be able to answer all the questions around it. So primary oocyte divides, okay, primary spermatocytes divide into two. This is meiosis 1 and you get secondary spermatocyte. Over here the same division happens, but you know what, it is unequal division. One is first polar body and the other one is secondary oocyte. So basically this does not divide further, only this will be dividing further. Now here this will undergo meiosis 2, give rise to 2 cells. Here also it will undergo division and give 2 cells. So at the end you have 4 spermatids. How many spermatids? 4 spermatids. But let's see what's happening over here. It is again going to divide into 2, but again unequal division. This is second polar body and this is going to be the ovum. This is going to be the ovum. So from one primary oocyte, how many ovum? You got only 1 ovum. From one primary spermatocyte, how many spermatids? You got 4 of them. So here 50 secondary oocyte is mentioned. 50 secondary oocyte will give rise to 50 ova, my dear children. Okay. And what about 50 secondary spermatocytes? 50 secondary spermatocyte will be giving what? How many? So each spermatocyte divides into 2, my dear children. So if there are 50 of them, so 50 into 2 will be what? 100. So 100 sperms will be produced and 50 ova will be produced. So your answer should be A. Your answer should be A. Oh my God. Okay, got it? Ma'am, please speak louder. Okay, Chirag, I will be coming to that. Okay, I'll be coming to that. Just give me a second. Hello, Surbhi, can you send Akash? Yeah, yeah. Okay. Okay, so until then I'll try to explain. Whoever can hear, listen to me, and whoever cannot, please wait. Someone is coming to help me out. So, oogenesis is basically you have got oogonia. Some oogonia they differentiate into primary oocyte, that is this, okay? And primary oocyte is going to undergo meiosis. So, meiosis happens in two stages, meiosis 1 and 2. Okay, so over here when meiosis 1 happens, you have got two cells. This is unequal division. Okay, so this one goes off, it does not divide further. This is secondary oocyte, only one secondary oocyte you will get. Again it will divide, you get one ovum because another division is also not correct. Okay, so this is about oogenesis. So from one primary oocyte you just get one ovum. Akash, they are saying that there's some issue with the mic. They're hearing some beep sound. Some children are, some children are not. Can you just check it out? Remove the mic. Okay, yes. Children, by then I think you can try solving this question. What happens when estrogen binds with oxytocin during parturition? Estrogen is with oxytocin. This becomes active and estrogen is there. Fine now. Let's see. Children, now are you able to hear? Audible? Am I clear? Yes? Can you give me thumbs up if you're able to hear me? Or give me a heart, something like that, so that I understand. Yes, 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 yes. Okay, so for I see certain children are not able to hear. Still there is a beep sound. Please check it out with your earphones, because I see most of you giving me hearts and thumbs up as well. Okay, so please check it out. Now we were over here. Consider the following statement with reference to female reproduction system. Statement 1. The presence or absence of hymen is not a reliable indicator of virginity or sexual experience. What do you think so? Is it a reliable indicator? No, it is not a reliable indicator. Why? Because hymen can be broken off even during high physical exercises, cycling, horse riding and things like that. So it cannot be a reliable indicator of virginity, and it should not be, right? So this statement is correct. It is not a reliable indicator. Now, statement number 2. The sex of the fetus is determined by the father and not by the mother. What about this? Is it true or false? Is it true or false? The sex of the fetus is determined by the father and not by the mother. Exactly, it is determined by the father because father are heterogametic. They have two type of gametes. Mother just have X chromosomes. Okay, so suppose from mother X chromosome came, but here X chromosome, so it's a girl child. From here X, but here Y, so that's a boy child. Actually, it should not be like that. Okay, both of them do not have anything to do, but if it is genetically, then it will be correct. Sex of the child is determined by the father. This is also correct. Both statement 1 and 2 would be correct. So C is the correct answer. Okay? Okay. C is the correct answer. Now, my dear children, I am going ahead with the next question. No, hymen is not a reliable indicator because it can be torn during any physical activities. Okay. So going ahead to the next question, that is reproductive health. Shall we? Okay. So I'll just explain parturition for someone who was asking about parturition, and then we'll go ahead. Okay. That is this. Listen to me. This is the uterus. Here you have the myometrium. Yeah. This is the fetus, fully grown fetus. It tries to move down. When it moves down, the cervix over here you have stress receptors. They become activated. Stress receptors active. Now they will be sending signals to what? Maternal pituitary. Maternal pituitary. And apart from that, one more thing is happening. The placenta which is here, my dear children, the placenta is also releasing too much amount of estrogen. What does this estrogen do? This estrogen will make certain receptors of oxytocin really active. What receptors? Oxytocin receptors. Now for maternal pituitary, when oxytocin comes, it gets an area to come and bind. So it comes and binds over here, and when it comes and binds over here, this is what the muscular layer, the smooth muscle layer, it will make it contract, and hence the baby would be moving down. This is what I wanted to tell you. Okay. Ma'am, is tertiary follicle and Graafian follicle same? It's not clear in NCERT. No, it's not at all same. Tertiary follicle is small, okay. Graafian follicle is very big, and inside the Graafian follicle, what do you have? You have secondary oocyte. You have what? The secondary oocyte, and the secondary oocyte has made up a new membrane, that is zona pellucida around it. Okay, chalo. Now, 42nd question. Which of the following cannot be detected in a developing fetus by amniocentesis? What is something that cannot be detected by the developing fetus by amniocentesis? Okay, Down syndrome, jaundice, Klinefelter syndrome, sex of the fetus. Shashank, yeah, I will be doing that. After reproductive health, we can do. There are only 6 questions, okay? So, all of you are saying B. The answer is B. Why B? Why B? So, basically, what is amniocentesis? You have the fetus over here, and it is surrounded by amniotic fluid. It is surrounded by what? Amniotic fluid, my dear children. So, in amniotic fluid, what do you have? The baby's certain cells also sheds down. So, when we are taking the amniotic fluid, the cell also comes together. In the cell, you have the nucleus. In the nucleus, you have the chromosomes and everything. So, basically, with the help of this chromosome, you can try to find any genetic disorders. And also the sex of the child. But the second part was used more often, and it led to female feticide. When people came to know that the baby is a female, they used to kill them. So that's how amniocentesis was banned. But it was initially made possible. Why? To check on any genetic disorders such as hemophilia, such as sickle cell anemia, and things like that, and Down syndrome. Okay, and jaundice is not just a normal disease. It's not a genetic disorder, which is why I'll say B is the answer. Okay. Okay, chalo, let's go ahead. Next question. Given below are 4 methods A to D and their modes of action. Okay, you have to do the correct matching the following and tell me. Sujata, I will be explaining, okay? I will be doing that. Mendelian disorders could be, all the genetic disorders probably could be got through with the help of amniocentesis, okay? Your genetic disorders, whatever it is. Okay. Chalo. So, you are saying C as the answer. Let me see if the C is the correct option or not. So, according to C, A, the pill goes with 3, prevents ovulation. Yes, it prevents ovulation. Why? Because the pill is a combination of hormones, progesterone and estrogen, and that basically have an imbalance in our system and does not allow ovulation to happen. So that is absolutely correct. Okay, that would be absolutely correct. Now going ahead with the next thing that we have, condom. Condom is going to act like a physical barrier. It does not allow gamete transport to happen. So I'll say C also goes with 4. C goes with 4, and P goes with 3. Okay, the pill goes with 3. Now vasectomy is basically when you cut off the vas deferens, right? So transportation of the sperm is not going to take place, my dear children. Again, it is going to cause trouble in the gamete transport. Now copper T, what it does, it's a very funny thing that copper T does. Copper T basically, you know what, it holds back the sperm. It does not allow the sperm to move properly. So it reduces the, what do you call it as, it reduces the motility of the sperm and also reduces the fertilizing capacity of the gamete. It reduces the motility of the sperm and reduces the fertilizing capacity, which is why your answer would be this. I hope that is correct. Okay, ma'am, in match the following, can the question have more than one answer, as in 43rd question in option C? 43rd question now. Yeah, it can have, okay, and it is, you have to be really careful for understanding that. And all of you who got it correct, my dear children, you are doing an awesome job out there. Chalo, it's already 154 children who are there. I hope all of you are doing great. Hello Shaila. Chalo, let's go ahead with the next question. CDRI stands for, this is a very easy question, but I put it over here because we forget the basic. Okay, Otaku Central and Rata Kumar, all of you are saying about the voice. Can you please check it, because I see you can hear, and also my team, okay, they can also hear. So I suppose there is some problem internally. Can you please check it out? That will be really kind of you if you do that. Okay, so all of you are saying option C as the correct one. It is correct. CDRI stands for Central Drug Research Institute. Now, who all have read NCERT properly will tell me the drug that they have given. Can someone tell me the drug that they have given? It is there in Lucknow. Great. UI Daniel, awesome. It is there in Lucknow. Can you tell me the drug? Jim Jam, Amog, Archana, status. Okay. Hi, Madiha, every one of you, Sania, Shaila, all of you, Kirtana, Saheli was the name of the drug. Now, my dear children, this gives you an important question. Is Saheli a steroidal drug? Does it cause a trouble in you? Saheli, is it a steroidal drug? Saheli is a non-steroidal drug. Remember that. Non-steroidal means you do not have any hormonal injections inside your body. Such as in pills, basically you are messing up with the hormones, right? You're giving extra hormones or doing like that. But in Saheli, it is non-steroidal. How does Saheli work? Basically, it goes and it blocks the estrogen receptors. It blocks the estrogen receptors. Now, estrogen, if it's not there, do you think endometrium can be formed and ovulation and everything will be happening? No. So that is how Saheli works. But it is non-steroidal. Please remember, it's very important. Okay? Once a week pill, exactly. Now, going ahead with this, chalo, let's identify what is what. Identify the figures of contraceptive given below and select the correct option that you have over here. So, I'll tell the easiest one to identify, okay? The easiest one to identify is this, that is implant. Implant, you can put it under the skin, my dear children. So, B is implant, and there are two options with that. So, we can cut off the other two options. Let's cut off the other two options. Okay, what about this? This is also very easy. Looks like a T. So T is what? Copper T, my dear children. Again, you have two options. Let's go ahead. Over here, this is the condom for males and this is the condom for females, my dear children. It has two knobs over here. Over here, just one knob. Fine. So your answer would be like what? Your answer would be what? My dear children, condom for female, condom for male and condom for female. This becomes your answer. D is the correct answer. So you all got D. Awesome job. Okay, chalo. Going ahead with the next question. We have administration of progesterone, estrogen combination of IUDs are effective within hours of coitus. How many hours of coitus? Can you tell me? Administration of progesterone, estrogen combination of IUDs are effective within hours of coitus. How many hours? 72 hours. Yes, exactly. So in case of rape or unwanted sexual activity, my dear children, it is given. Why? Because within 72 hours, you know what? Sperm, when it enters the body, it can stay in the body for almost 5 days. Okay, almost 5 days. So basically, if we want to kill those sperms, we can give these drugs, okay, such that the sperm is not killed, but anyways, ovulation would not be happening and implantation would not be happening because the endometrium line will be very thin. And that is what we want. Okay, that conception should not be happening. So it is given within 72 hours. Always remember that. So all of you who said A is the correct answer, you are absolutely doing a fantastic job. Now 47th, this question is tricky. This question is tricky. Please tell me the answer properly, okay? let's see. So over here, medical termination of pregnancy, MTP, is considered safe up to how many weeks of pregnancy? How many weeks of pregnancy? Okay, so I see a lot of people says A, B, 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 B. Okay, 8 weeks, 12 weeks, 18 weeks, 6 weeks. Your answer is what? 12 weeks. Up to 12 weeks, it is considered to be safe. Now I'll ask you one more question. Okay, I am going to ask you one more question. You have to tell me. Until which month, okay, or until which week can someone perform MTP? I'm not asking about safe. I'm not asking about safety concern. I'm telling doctors allow MTP until which week? 24 weeks. So who is that person who gave the answer? 24 weeks. Amma, that went. Well, someone gave the answer 24 weeks. Now it went. Anyways, Monica, Pavan, okay, all of you, my dear children, the answer is 24 weeks. So until that, you can perform MTP, but it is safer up to 12 weeks. It will be safe up to 12 weeks. So please remember that. Okay. Chalo, going ahead. The following factors indicate improved reproductive health of the society. Choose the correct option. Can we choose the correct option, my dear children? Okay, you have to see improved reproductive health of the society. So better detection and cure of the disease. Does that improve the reproductive health? Yes, obviously. Better postnatal care, that means after the baby is born, after that better care, yes, that also gives a good thing. Medically assisted deliveries, obviously yes. Increased MMR, what do you mean by MMR? Maternal mortality rate. So if mothers are dying more, do you think it is reproductively healthy? No, it is absolutely wrong. So increased MMR is a wrong thing. It should be decreased MMR. Okay, so I'll say A, B and C is the only thing. So A, B and C comes in what? B option. So this becomes your answer. Okay, all of you? Understanding? Maternal mortality rate. Yes, Pawan. Awesome children. Awesome. I see so many of them. I think we can make it. What do you call it? 200 this time. Anyways, I am going to explain you something very important, and that is menstrual cycle. Listen to me carefully so that you can answer every question related to it. Okay? Menstrual cycle. How does it work? So we have to divide it into 3 phases. That is menstrual phase. Menstrual phase. I'm not going very deep. Something that is important, that is only I will be discussing. Okay, then you have proliferative phase, and you also call this as follicular phase. Okay, next we have, after that what do we have? Yes, after that we have luteal phase, and also secretory phase, okay. Now listen to me, what happens? Suppose fertilization didn't happen, okay? There is no fusion of egg and the sperm, which is why our body cries with blood, my dear children. So there is shedding of endometrium. There is what? Shedding of endometrium. But then it happens from 1 to 5 days. Okay, almost from 1 to 5 days it will be happening. But then our body thinks that, okay, let's give it one more try. Okay, let's give it one more try. Slowly what will happen, my dear children? The next set of follicles, they start maturing. So in follicular phase, the follicles are going to mature, and one follicle will be growing into a mature Graafian follicle, having what? The secondary oocyte. Okay, now when the follicles are being maturing, you know what? It is made up of granulosa cells also. These granulosa cells are going to release something very important, and that is estrogen. And who is making these follicles mature, my dear children? It is FSH and LH. Majorly FSH, follicle stimulating hormone, that will be making these things mature, and also LH to a certain limit. Now these follicles are in turn releasing estrogen, and what will happen because of the estrogen? Slowly the broken endometrium will start reforming. It will start reforming, which is why it is known as proliferative phase, and this is why, since follicles have been made, it is known as follicular phase. Now when the amount of estrogen is released way too much, what will happen? All of a sudden, because of this estrogen spike, my dear children, all of a sudden the LH will also increase way too much. There is an LH surge which happens. Because of which the Graafian follicle becomes scared and it breaks open, my dear children. When it breaks open, what are we left with? Basically, secondary oocyte is released. Apart from that, everything that is left over, it is known as corpus luteum. What do you call it? corpus luteum. Now who maintains the corpus luteum? It is the LH only, because LH did all the thing. Okay, so it is known as luteal phase because corpus luteum is being made. Now corpus luteum, my dear children, it will make two things. One is progesterone, and the second one is again estrogen. It will make it. Okay, it will produce it. Because of progesterone, my dear children, what will happen again? This endometrium will become more thickened. It will become more thickened and will also start secreting certain things. Why? Why does it do so? Because, my dear children, what happens when the secondary oocyte is released? I believe the body is believing that anytime fertilization can happen, anytime embryo can be formed, and I have to provide nourishment, which is why the progesterone start releasing what? Secreting glucagon and things like that, so that it can provide nourishment. So this is what happens. Now the story reverses, my dear children. When the progesterone is being released, the progesterone says that I do not want to see LH and FSH. I do not want to see LH and FSH. So basically, it tells the hypothalamus to stop releasing GnRH, gonadotropin releasing hormone, which was the hormone which is telling the pituitary to release these two things. So when GnRH tells the pituitary stop release of FSH and LH, what will happen? Without LH, do you think corpus luteum will be there? No, corpus luteum is not going to be there. If corpus luteum is not going to be there, do you think progesterone will be there? No, progesterone will also be not there. In that case, do you think endometrium will be maintained? No, it will be slowly shedding down. You get it? So this is thing that happens again and again. Now suppose fertilization happened. Okay, suppose fertilization happened. Same thing, the progesterone will still go and say GnRH to stop. But there will be something else that is HCG, human chorionic gonadotropin, that will be maintaining the corpus luteum to have repeated secretion of progesterone. Okay, I hope now you got it. So basically, on this particular day, that is almost middle of the cycle, suppose my cycle is of 28 days, middle of the cycle, that is 14th day, there will be what? Ovulation. There will be ovulation. Works? Okay, on the 14th day, ovulation would be happening, and rest of the days, the same thing goes on the 28th day. After the 28th day, from 1st onwards, again, the menstrual flow will go on. Did you understand it? So, 1st to 14th is proliferative. No. 1st to 5 to 7 days, you call it menstrual phase. Then from this to 14th, you call this as proliferative or let's say follicular phase. And then from this 14th to 28, you say luteal phase. Okay. I'll tell you something, the dates is not going to be happening. Question. You would be asked, what is the fertile period? For a woman, what is the most fertile period, my dear children? This period. That is almost 1 to 7 days where it is happening. 1 to 7 days of this. Okay, sorry, I will say 10 to 17th. Suppose this is a 28-day cycle. 10th day to 17th day, it is fertile period because any time ovulation can happen. Okay, which is why it is a fertile period. And here comes the role of. Please remember, it is 10th to 17th, not 12th, nothing. 10th to 17th. 7 days, fertile period. So you might have heard about natural way of avoiding conception. Okay, in that the first one is what? Periodic abstinence. What do you mean by that? You are not performing sexual activity during the fertile period. That is periodic abstinence. Okay, that is natural contraceptive method. Second one under natural contraceptive method is withdrawal mechanism, where you are withdrawing the penis out of the female genital tract. Okay, before insemination. Now the third thing is lactational amenorrhea, where a mother who is lactating, would not be producing the egg. That is, she would not be ovulating, my dear children. But is it safe? Listen to me, there's a very important question. Do you think this is safe? Can you rely on it? No, it is not at all reliable. Please understand, natural methods are not at all reliable. Then comes physical barrier. You have condom, and you have diaphragm, you have vault and everything. It becomes more effective when you use spermicidal creams and things like that. Okay, children, I am revising. I hope you're getting it. Now, the third thing is IUDs, intrauterine devices. They are the best. Why they are the best? Because basically, they help in spacing the children, and also it is most widely accepted technique. You have 3 types of IUDs. One is basically non-medicated IUDs, such as lipase loop. Such as what? Lipase loop. You insert it in your body feels it's foreign and basically reacts to it. When it reacts to it, anything that comes along with it, for example, a sperm also, it will be killed. So that is how non-medicated IUDs work. Example is lipase loop. Second thing, okay? I'll come, okay? Vishwa. I'll come to questions. Second thing is what copper releasing IUDs. Now I'll make you participate. Tell me certain names of copper releasing IUDs. I'll tell you the concept behind it. So copper releasing IUDs, my dear children, they are going to release copper which is going to mess up around the motility of the sperms. They would not be able to move around. Their fertilizing capacity also decreases. So CU7, yes, you're getting it correct. CR Shetty, CUT, Monica, CU7, CUT. I want one very important name which confuses everyone. Multi-load. Multi-load 375. Yes, exactly. It's multi-load 375. So it's like 375 units of copper. That is what it means. Okay, multi-load 375. CU7 example of copper releasing IUDs. Now you have the third one, hormone releasing IUDs such as Progesta Cert. And what? And one more. Tell me the example of hormone releasing IUDs. So how does it work? In your body, progesterone and everything is released normally. But if you're giving externally also, the rhythm gets different. Okay, and when the rhythm gets different, my dear children, what is going to happen when the rhythm gets different? It is going to create a huge trouble. Basically, a woman who was able to ovulate would not be able to ovulate. The endometrium also gets very thin. And also one more thing happened. The cervix, okay, the uterus is known as, there will be a cervical mucus plug being formed so that the sperm cannot get inside. Okay, got it? So all of LNG20, you're absolutely correct, my dear children. LNG20 and progesterone, they are hormone releasing IUDs. And then after that, what do you have? Yes, cervix becomes hostile. Ma'am, my voice is breaking. Is it fine now? Is it fine? And okay, everyone says voice is breaking. No? Now fine. Okay. Ma'am, voice is breaking if you move. Oh my god, that is a very... Okay, I'll do something else. So, is it fine now? Okay. Samprita says yes, everyone else says no. Okay. Wait. Wait, okay. Children, yeah, now my voice is clear. I will talk about that the person needed, but oh my god. Okay, children. Okay, children, I think it's fine now. No, it's fine. Give me a second, okay? Give me a second. I'll be right back. Yeah, fine. children. We are working on it. Can we change this from that studio? Okay. Every one of you, can you hear me now? There's a new mic. Please tell me you can hear me now. Okay, perfectly fine. Thank you so much. I think there was crystal clear. Great. Chalo. So I was here. We were talking about pills. Okay, pills also works on the principle of hormones. Okay, progesterone, estrogen, similar thing. Okay, so you do not have to take pills when you're menstruating. That is 1 to 7 days. Apart from that, all the days pills have to be taken. If it's Saheli, it's a once in a week pill and it's non-steroidal, none like the other pills. Okay, now let's go ahead with the next question. Starting off from this particular chapter, my dear children, so sorry for keeping you waiting. It's biotechnology, principles and processes, very important chapter. So mechanism of pills, Abdija, as I said, my dear child. So it works on the principle of hormones. Okay, works on the principle progesterone and estrogen. Basically, if you're giving it to your body from outside, it imbalances the hormones, which is why ovulation does not take place properly. Endometrium lining is very thin, and also cervical mucus forms. Cervix becomes hostile. Cool. Now, fine. You are answering this question, I suppose. 49D, someone says TI plasmid is useful for bringing genes into animal cells, bringing new genes into plant cells, bringing tumor cells into animal cells, and all of the above. What will be your answer for this? What will be your answer for this? So I see B, C, D, all the options out there. What is the correct answer? Let's see. It is for bringing tumor cells in plant cell, is it? What do you think so? No. You are asked what it is useful for. So generally TI plasmid, okay, generally TI plasmid has a big role to play. So it belongs to agrobacterium tumefaciens. In agrobacterium tumefaciens there is a TI plasmid, that is tumor inducing plasmid, which has a part which is known as tDNA, transfer DNA. So basically when a plant is infected with this particular bacteria, it transfers the tDNA part, causing tumor. But we are very intelligent people, right? So what did we do? We use the same mechanism, and what are we going to do? We are going to replace this tDNA with our gene of interest. We are going to put our gene of interest, and now go and infect. When it goes and infects, my dear children, is it actually passing down the tDNA? No, it is passing down the gene of interest. So it is perfect vector for plant cell, because agrobacterium tumefaciens affects the plants, not the animals. Please remember that. So which is why your answer becomes B, bringing new genes into the plant cell, not C. Okay, not C, it is B. So your answer is this. Going ahead with the next question that you have, please be very careful while solving this question. I also messed around it. Okay, so the figures shown below, A, B, C step are there. A can be at multiple places, B can be at multiple places. So you have to be really careful with this question. Okay? Chalo. Someone says favorite chapter, Chandrakanta. Yes, mine as well. So what is the ratio of test cross in monohybrid? Ramya will come to that, okay, later. Now, children, asking numbers and all, we'll do it at different time. So A, Neeraj says A, Amok says D, Neeraj says A. Let's see what is the answer for this particular question. Okay, so over here, A should be either denaturation. A should be either denaturation. So A here is annealing and denaturation at temperature of 50 degrees Celsius. 50 degrees Celsius, do you think denaturation can take place? No, my dear children. Denaturation needs up to 90 to 92 degrees Celsius. So this is wrong. This statement is absolutely wrong. Now, so this is also wrong, because A is not annealing. Okay, annealing means you are adding primers up there, and A over here is there, it is not annealing. A over here is also there, it is elongation but not annealing. So those two options are wrong. Now you have C. C is extension in the presence of heat stable DNA polymerase. C, are you extending? No, C is basically annealing, where you are putting the primers up there. Okay, that is C. Over here, extension in the presence of heat stable DNA polymerase. So here also C is mentioned. Now B is mentioned over here. What is B? B is this, okay, and B is denaturation at higher temperatures separating two strands. Denaturation at higher temperatures separating what? Two strands. Can this be B? Yes, this can be B, and even this can be B, my dear children. So what is your answer for this particular question? Ayyo. Yes, there are on two sides A, B, C, so you have to be really, really careful with that. Okay. So it is D, it is D, dear. Abhijit, Samuel, it is D, but there is an option with C also. There's a clinical thing with C also. C is basically extension in the presence of heat stable DNA polymerase. So this is C, where extension is happening actually. Okay, in the presence of heat stable DNA polymerase, and B is denaturation at higher temperatures separating the two strands. That is also correct. That is also correct. So I'll say C and D, both the options are equally correct. Okay, this option will end within soon. It's like 10-15 questions are left. That's all. Chinmay. Works? So both these options could be correct. Fine. Chalo. Going ahead with the next question. Identify A, B, C and D in the following diagram PBR 322 and mark the correct options given below. Okay, mark the correct options given below. Let's see. So what is A over here? So I'll tell you the easiest thing to find out. The easiest thing to find out is B. B stands for what? Origin of replication. Origin of replication. So only one option has B and this becomes your answer. Only one option has B and this becomes your answer. What is it? B option. So B over here is origin of replication. What is D? Let's see if it's correct or not. D is the ROP, the replication protein that we need. That is also correct. Now A is what? The tetracycline resistant gene. Over here there's a gene for tetracycline resistant. Okay, and over here C. What is C? Ampicillin resistant gene. So this is how PBR 322 is being designed. So your answer becomes what? B. So who all got B? How to identify, ma'am? You have to study it. Always remember. Okay, always remember this is how it goes. So here you will be having origin of replication, where the DNA polymerase will come and bind and do all the things. Here you will be having ROP, okay, just near to it. Now always remember both the sides you will be having a selectable marker, that is a resistance gene. On this side it will be ampicillin, on that side it will be tetracycline. Fine. Now everywhere here you will be having two sites for enzyme. One is for BAMH1 and one is for SAL1. On the other hand, here also you have for two. One is PVU1 and the other one is PST1. Okay. Now here at the top 2 and 2, and then here at the top you have 3 of them. One is EcoR1, second one is CLAV1 and the third one is HIND3. Third one is what? HIND3. Okay. And now last, my dear children, over here also in the ROP site also you have one, and that is here you had PVU1, here you would be having PVU2. This is how you answer. Okay, works? Shall we go ahead? 52nd question, try it out. Could you explain the diagram? If we talk about the diagram, okay. My dear child, there is actually nothing much to explain. So listen, this is where the DNA polymerase will come and attach. ROP is a very important protein for replication to happen, so ROP site is there. Now this site is ampicillin resistant site. That means if I try to grow the organism containing this plasmid in ampicillin, it will definitely grow. Why? Because it has ampicillin resistant gene. Similarly, it has tetracycline resistant gene, so I can grow it in a medium that contains tetracycline also. But suppose I am using PVU1 enzyme, okay, and trying to cut this particular plasmid. If I am using PVU1 enzyme, it will cut over here and it will insert the thing over here. If I am inserting something over here, do you think it will still be resistant to ampicillin? No. So this is how question comes. So it will be said that you are inserting something. Will it be still tetracycline resistant? No, it won't be. So you have to know where is the site located. BAMH1 and SAL1 site is here. Okay, PVU1 and PST1 is over here. EcoR1, Clavin and Hind3 is over here, and PVU2 is over here. If PVU2, if I am cutting it over there, what will happen? ROP protein is not going to be produced. Cool? Next question. A plasmid has two antibiotics. Yes, here comes what I was talking about. Okay, here comes as I was talking about. So that is, a plasmid has two antibiotics resistant gene, one for ampicillin and one for tetracycline, just as we saw. It is related with a restriction enzyme that cuts in the middle of ampicillin gene. So there is some restriction enzyme that has a spot in the middle of the ampicillin resistant gene. Now DNA fragment contains a hemoglobin gene where cut with the same enzyme, same thing, okay. The plasmid and the fragment were mixed, treated, and ligase were used to transform the bacterial cell. Clones are kept. Now you have got a recombinant DNA. In short, you have got a recombinant DNA where what has happened? In the ampicillin site, you have inserted something else. Now you tell me, it can grow on plates with both antibiotics, that is ampicillin and tetracycline. Can it? Can it grow on both? Are they resistant to both? Are they resistant to both? No, it is not. Okay. Can grow on plates with ampicillin but not with tetracycline. Array, it can grow in tetracycline but not in ampicillin, because in ampicillin you made a cut. Now it is not resistant anymore. So C is the answer. Can grow on plates with tetracycline but not with ampicillin. Now cannot grow with any antibiotics. That is wrong. So C becomes your answer. Okay. So all of you who who understood the question, can you just give me a thumbs up or put me a heart? Something like that, so that I get to understand that everything is clear. Yes, thank you Ratan Kumar for giving me a thumbs up. Aisha, Meera, Shan, okay, Shanmuka, Abja, okay. Thank you children. Chalo. Next question. Which restriction enzyme must be used to cut the DNA given below? Okay, I'll read out the names now. Bhagya, Sanjana, Nikhil Gowda, TV Venkatesh, Shashank, Trio, JE student Ibani, Karthik, Neeraj, Swapna, Chandrakanta, Nikhil Gowda, Chandu, Jim Jam, Rashmi, Baswaraj, Saumya, Chirag. Hey. Come on guys, be good children out there. Nikhil Gowda, okay. Hint 2, someone says hint 2. Someone says it's B. Chalo, let's see who will got the answer. The answer is B. Why do I say B? Okay, so over here in the question it is clearly mentioned which restriction enzyme must be used to cut the DNA given below. And in the DNA you see the site 5GAATTC, 5GAATTC, this side also, this side also. So don't you think GAATTC site is for what? It's for EcoR1. How did you forget that? GAATTC in 5' to 3' direction. So it's EcoR1, it can make a cut over here and also over here between G and A. So your answer becomes what? B. Always remember, HIND2 was the first restriction enzyme to be found out, and in the book it is written it has 6 base pairs. Okay, it has base pair, 6 base pairs as a restriction site. In EcoR1 also you have 6 base pairs, 1, 2, 3, 4, 5, 6. But it is EcoR1's thing is given. Okay, I hope it's clear. Okay, chalo. Next question, 54th. Which of the following step is not involved in the procedure of making host cells competent? Competent as in making capable of, taking in the DNA. Taking in the DNA. So all of you are asking, explain other options. So HIND2 is ALU1 has a different restriction site. HPA1 has a different restriction site, my dear children. In your NCERT, you just have this option, so go with this. Okay. Okay, now try it. Now try this out. Ma'am, when will it end? Faizan, it will end soon, my dear child. It will end soon. So we are in 54th question. It is 66 questions are there. I mean 11 more. 11, 12 more. Yeah. Okay, A, D, everything is coming in. So which of the following step is not involved is asked. So treating bacterial cells with specific concentration of divalent ions. Do you do that? Yes, you do that. Why do you treat it with ions, calcium and things like that, my dear children? Basically, your DNA is basically what? Negatively charged. So you want to neutralize the effect so that it can easily pass the membranes of the things that you are sending it into. So that is why you treat it with divalent cations. Now that is needed. Now incubation of host cells with recombinant DNA on ice. Do you do that? Do you incubate the host cell with recombinant DNA? This is the host cell, suppose, and this is the recombinant DNA. Do you put it inside ice? Yes, you put it inside ice. This is also correct. Why? I'll tell you. Okay, so this is cool. Okay, the temperature is very stable. The cell membrane is very stable. Now, after that, you will give a heat shock. That is, you rise the temperature suddenly, and when you do that, when you shift from ice to heat condition, there will be transient pores, as in temporary pores out there, and with the help of this temporary pores, what will happen? The DNA which has neutralized content, my dear children, it will slowly get inside. It will slowly get inside, and now we want to close the pores. So what do? We again put it back into the ice. So incubation of host cell with recombinant DNA on ice, that is correct. Heat shock at 42 degrees Celsius and putting it again on ice, that is also correct. Dissolving cell wall with enzyme, that is wrong. Why do you want to change the host cell? Should not be changing the host cell. So your answer becomes D. So who all got D? You are doing absolutely great. Who all didn't get D? It's totally fine. We make things correct later on. Yeah? Are these questions enough to get Jeffia and John? I just hope so. Yes, I'm trying my best to make you get more than 45. Yeah? Children, Chirag and everyone out there, Soumya, Chirag, children, let's be good children out there. Yeah, let's be good friends. Okay, question number 55. It is useful for NEET as well for understanding your concepts. Yeah, which statement is incorrect? Retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. Do you think? Children, answer. Retroviruses have been disarmed, have also been disarmed and can be used to deliver desirable genes into animal cells. Yes, it can. For animal cells, my dear children, you specifically need retroviruses. Always remember that. Now, downstream processing is one of the steps of our DNA technology. Yes, downstream processing means purification and things like that. It is a step of recombinant DNA technology. Now, DNA is negatively charged molecule. Yes, no doubt about that. Presence of chromogenic substrate gives blue color colonies if the plasmid in the bacteria does not have an insert. Is that correct? Is that correct? So basically it says that there is, suppose a plasmid, okay, and this plasmid has a gene, that is LAG-Z gene, which codes for beta-galactosidase. Okay, now when I put X-gal, that is a substrate, I put a substrate, so what will happen? This beta-galactosidase will react on it and will be giving a blue color. Normally. But suppose I am cutting this gene off and I am putting my gene of interest. Will it still give me blue color? Array, it's not being produced. How can it convert it to blue color? So it won't give me any color. It will give me white color. So the presence of chromogenic substrate gives blue color colonies if the plasmid in the bacteria does not have an insert. That is true. If it does not have an insert, it will give the blue color. Right? So this is also true. So can I say all the statements down there are true? All of them are true, my dear children. All of this are going to be true. Do you have any other answer for this? Okay, which of them is incorrect? That is being asked, right? So none of this, none of them are going to be incorrect. This becomes your answer, D. Got it? Okay, cool. Chalo. Next. 56th question. With respect to DNA fragmentation, you have to tell the answer. Statement A says, gel electrophoresis and elution are two important processes. Okay, Chinmay, Baba, we will do it tomorrow. Not tomorrow, day after tomorrow. PU1 concepts will also be there. Tomorrow, chemistry and physics. Okay, with respect to DNA fragmentation, statement A, gel electrophoresis and elution are two important processes. Yes, it is. Gel electrophoresis means you are running the DNA on a chamber, okay, and is moving from negative end to positive end. That is true. And elution is basically when you are cutting down the DNA from the gel. That is elution. So both of them are important. Statement B, after staining with ethidium bromide, it has to be exposed to UV light. Is it? If you want to see the DNA, you have to stain it with ethidium bromide, and then you have to expose it with UV light. Then only you will be seeing it. And what is the color? Can anyone say color? Can anyone say color? What color it will be there? Arjun, I took your name. Chalo, fast, tell me the color. Yes, it will be bright orangish color. The bands will be appearing in bright orange color. So can I say both A and B are correct statements? Yes, I can absolutely say that. Okay, and with that, we end the chapter. Now, my dear children, I want few questions answered. Okay, not there in this particular thing. Fast, okay. I want to separate the DNA out. I want to separate the DNA out of like in a test tube. What is something that I will add for separating the DNA out? What is that something that I will be adding for separating the DNA out? Fast. This process is known as pooling. This process is known as pooling, where you take out the DNA. I am asking you, how do you separate the DNA? Chilled ethanol, Reuben, great. Ibani, great, my dear children. Vaishnavi, Rajita, all of you are doing superb. Anuke, so it is chilled ethanol that I will be adding. Now, one more question to you guys, okay? DNA would be moving from which to what direction to what direction? Cathode to anode, or anode to cathode? What is your answer, A or B? DNA, when you put in gel electrophoresis chamber, in which direction do they move? They move from cathode to anode, anode to cathode. Let me see. So I see Akshata gave me the correct answer. Okay. Someone asked me to drink ethanol. I thought you guys loved me. Okay. Okay, Kushal, I see all of you who are attempting it. Sahana, Kushal, Chirag, my answer is not correct. Okay, listen, it is cathode to anode. Why? Cathode, my dear children, cations are positive but cathodes are not positive. It is negative, cathode. So our DNA is also negative. Negative, do you think it can stay together? No, it will move away. It will move to the anode. Anode is what? Positive. Okay, anode is positive. Always remember, cats have pause. Cats have pause. So cations are positive, but in case of cathode, it is opposite. Okay. Works? Ratan Kumar, your class is superb. Thank you so much. Okay, so I hope that is clear. Apart from that, any more questions that I want to ask you guys is insertional inactivation. I hope you know that. Insertional inactivation, basically what happens when you insert something and because of that the function is lost, you call this as insertional inactivation. Okay, works? Chalo, let's go ahead. Abdija, thank you dear. Okay. Health, human health and diseases, can we start that? Are you ready? Chalo, ready. Ready. Do we have, okay. That is the last chapter and with that we are going to finish. I want to hit 200 now. Okay, in at least this particular last chapter. And all of you who are liking the video, please do not forget to like and comment it down. Chalo, first question, heroin. Heroin is what? A cannabinoid distylmorphin chemically, okay, commonly known as coke or crack, or used to treat mental illness like depression and anemia. Insomnia, oh my god. Ma'am, little bit slow, okay, I'll be a little bit slow now. I see 164 people watching the live, awesome children. Maurya, thank you so much. Okay, so B, C, everything, all the things are there. B, Jim, Jam, Shruti, Chandu. I'll try to read out your names as well in between, okay? So heroin, my dear children, it is what? Diacetylmorphin. It is not a cannabinoid. Cannabinoids is something that you get from cannabis sativa. Okay, and cannabis sativa produces what? Ganja, charas, marijuana, hashish and things like that. So heroin is an opiate. You get it from opium poppy or Papaver somniferum, okay? And when you do acetylation of morphine, you get heroin. You get heroin, my dear children. So this will be the correct answer. Commonly called coke or crack, wrong. It is known as smack. It is known as smack. Now, used to treat mental illness like depression and insomnia. No, my dear children. You know what? If you take heroin, It is a depressant. It will slow down your body very much. It is what? A depressant. Always remember. So I gave you all the features about heroin, so please do not forget it. Sedative is what? Sedative is morphine. So under opioids you have two things, morphine and heroin. Heroin is a depressant. Morphine is what? Sedative. That means it will make you sleep, and also it is acting like a painkiller. It is also acting like a painkiller. Just during surgeries you give this. Okay? Work? And where do you get this mark? Okay, where do you get heroin or things from? Basically latex of poppy. Latex of poppy plant. So in your book, image is given about the structure of what do you call morphine. Just remember it looks like O. So O comes under opiate. So morphine is opiate. Okay? Ma'am, when do you come to Hubli? Oh my God, I wish to. I don't know, but chalo. Let's start. Human immunodeficiency virus is comprised of a protein coat and genetic material, which is what? Which is what? Human immunodeficiency virus, HIV, is comprised of human protein coat and a genetic material, which is single-stranded DNA, single-stranded RNA, double-stranded RNA, double-stranded DNA. So Anu, Arjun, Monica, Ratan Kumar, Trisha, Chandrakanta, Nikhil Gowda, Hema, Chinmay, Swapna, all of you are there. Ibani, Pavan, Trisha, Samprita, Nandish, Pratibha, oh my God, Praniti, awesome children. So, my dear children, the answer for this question is actually B, single-stranded RNA. So, if we talk about the, okay, in the case of, what do you call, in the case of HIV, they have single-stranded RNA, but there are two of them. So, you have two single-stranded RNA, but it is not double-stranded. Remember, there are two of them. Two of them are there, but both of them are independent. Always remember that as well. So your answer is anyways B. Okay. Next, read the given statements carefully. Or you. Did I tell the answer? Okay. The answer itself is marked. But let's see anyways. Which of the above statement is correct? Innate immunity is specific type of defense that is present at the time of birth. Do you think innate is correct? Do you think it is correct? Yes. You have two types of immunity, innate and acquired. Acquired, you acquire it throughout your lifetime. Innate, you have from your childhood. So, it is specific type. But it is specific. Do you think it is specific? Are you wrong? Are you? It's wrong. So, the answer over here is also wrong. Okay. So, now this question is easy for you. Now, try out this question. This is not the answer. Not the answer. Children, is it specific? Innate is not specific. Okay. It is not specific. It is non-specific. That means basically innate would not be producing different things for different things. For example, if virus comes in, if any other microbes come in, your skin is skin. Does this change for everyone else? No. So it is like basically non-specific. So now everyone is saying A and D. Let me see. Now malaria could be confirmed by Vidal test. No. Vidal test is for typhoid. Malaria is caused by Plasmodium. So wrong statement. So this is also wrong. That is also wrong. Now, malignant malaria is caused by Plasmodium falciparum. Yes, the malignant form of malaria is caused by Plasmodium falciparum. Correct statement. Active immunity is low and takes time to give its full effective response. Yes. Why? Because in active immunity, you want your body to be actively producing antibodies. So, there is no help. Basically, you are just giving antigens, and that is obviously a slow process that to make antibodies. So, this is correct. Okay? This is correct. Now, saliva in the mouth acts as physiological barrier for pathogens. Is it a physiological barrier? Saliva in the mouth? Yes. That is also correct. So, I will say second, fourth and fifth is correct. Second, not third, fourth and fifth is correct. Third, fourth. There is no option like that. Ma'am, option itself are wrong. Let me see. Malignant malaria is caused by Plasmodium falciparum. That is correct. Active immunity is slow and takes time to give its full effective response. That is correct. Saliva in the mouth acts as physiological barrier. Yes, it's a for pathogens. That is also correct. So 3, 4 and 5 is correct. So yes, as you said, the options are wrong. Probably this should have been 3. So 3, 4 and 5 are correct answers. Rest all of them are wrong. Okay. So all of you who got this are correct. You are guys are also correct. Now going ahead with the next question. Natural killer lymphocytes are an example of natural killer. Yeah, 1 grace mark, you'll get it. Natural killer lymphocytes are example of what? Physical barrier, cytokine barrier, cellular barrier, physiological barrier. So my dear children, you have your innate immunity, you have different type of barrier in that. Okay. Hi Shreya. Okay, so natural killer cells comes under cellular barriers. You are absolutely correct. Okay, so physical barriers in your skin and everything. Okay, physiological barriers in your tears, your saliva, and HCl and things like that. Something that is being produced by your body. Cellular barriers as in cells that are produced. You have certain cells such as macrophages. Apart from macrophages, what do you have, my dear children? You have this thing. What do you call? Natural killer cells, NK cells. Apart from that, you have PMNL, that is neutrophils, polymorphonucleoleucocytes. So all of them, they come under cellular barriers. Now cytokine barriers are something that the cell releases and that acts as a barrier. For example, interferons. Okay, so they also come under that. Monocyte, yes, monocytes comes under this. So I hope that is correct, clear. My dear children, these interferons are for what? Interferons, which are cytokine barriers, as in the cell releases interferons. What do they do? Can anyone tell me? Hi, I am Harshita. Hi dear. Can anyone tell me fast? Alpha interferons. Normal interferons. I am talking just about normal interferons which are there. No one knows it? Come on. So basically, protect the unaffected cells. Exactly, Yashashwini. Listen to me. The other interferons, alpha interferons, are given in under cancer. That is basically going to act like a biological modifier. Okay, that is something different. Over here, normal interferons are the ones, basically if suppose this cell is affected by virus and this cell is crying, basically this cell is going to release something very, very important, and that is interferons. And what are these interferons going to do? They are going to tell the other cells that, hey, virus attack have happened, you have to be safe. So this is going to protect the other unaffected cells. This is the role of interferons. It is produced by virus infected cells. If the question comes, okay, you are given option, in which of the diseases do you think interferons will be released? And in the option, if HIV AIDS is mentioned, go for this because this is a virus infected disease. I hope this is okay to you guys. You got it? Chalo. Next, select the correct option showing the life cycle of Plasmodium. Now I want to get answer for this particular question. Let me read out your names. Hi Manasa. Ma'am, can you re-explain please? Trisha, which question da? Can you just tell me once? Ayo. Which one? 60th question. Okay, I will explain. Okay, I'll come back to that question. Once we finish this, can I? Is it okay for you? So those of you reading this, they can answer this, right? Okay. You're saying D is the answer for this question. Someone says D, someone says okay. So many different opinions are there. So select the correct option showing the life cycle of Plasmodium. Let's see. Sporozoites. So listen to me, okay? Listen to me and then try to find the answer carefully. Fine. So first thing, sporozoite is there in the mosquito's salivary gland. So it gives us. So in humans we have sporozoites. In humans now we have sporozoites. Now it's like the terrorist, and terrorist has two camps over here. First camp is liver. It gets into the liver, okay, and gets into the cell and basically bursts open the liver cells and goes outside. Where does it go? It goes to the second camp. What is the second camp, my dear children? RBC. It goes to the RBC. Now, in the RBC, what is going to happen? In the RBC, they are going to transform into different stages. One such stage is gametocyte stage. Gametocyte stage means you are going to form gametes next. But do they form gametes in the human body? No, they are not going to form gametes in the human body. In the RBC, what they are going to do? They are going to make gametes, and when the RBC bursts open, what will happen? These gametocytes are going to be there in the blood. But do they make the gametes? No. Now what will happen? Blood meal. Our mosquito guy will come and take this blood meal. When it takes the blood meal, my dear children, what will happen? These gametocytes are going to go in the gut of the mosquito. There, it will form the gametes. It will, fuse. It will fertilize and later form something else that is known as ookinete. From the midgut, from the gut of the mosquito, my dear children, this is going to transform into the next stage, that is sporozoite again. So sporozoite stage is again reached, this time where? In the mosquito's salivary gland. In the mosquito's salivary gland. Again this mosquito will come and bite us, and the same thing repeats again and again. Now you tell me the answer for this particular question. Eating mango, even I want to eat mango. Okay, so over here, let's see. So your answer is what? Your answer will be B. Sporozoites human, then it affects the liver cells, goes to the RBCs, gametocyte in the blood, then the blood meal, okay, by the mosquito, bite, multiply in the mosquito, sporozoite in the mosquito. Hence, your answer becomes this. Okay? Works. PJ Gaming video is too long, but if it has information, we should be definitely watching it. Right? Okay. 62nd question. 62nd question. Going ahead, enzyme which is responsible for replication of HIV in the macrophages of the host is. Enzyme which is responsible for the replication of HIV in the macrophages of the host is what? Pavan, yes, hemozoin is being released then. I'll talk about that as well, okay? It is what? Reverse transcriptase. So basically I told you HIV has RNA, and if it wants to get incorporated into our DNA, my dear children, it has to form DNA. So DNA to RNA, what do you call it? You call this transcription. Ulta of it will be what? Reverse transcription. And who helps that? Reverse transcriptase enzyme would be helping that. Hence it will go get into our DNA, and then what will happen? It will make copies out of it, my dear children, in the macrophage. In the macrophage, it is known as HIV factory. They will make so many copies. They are going to make so many copies. They are going to increase the number, and now they are going to affect which cells? Can you tell me? HIV viruses affects which cell? Even though HIV factory is macrophages, but they are going to affect some other cell. Can you tell me that? Anyone out there? So I see so many people answering from the very start. I'm really proud of you guys. Okay. It is what? T helper cells. Exactly, Madiha. T helper cells. So basically from here they are going to affect the T lymphocytes. Okay, and in T lymphocytes also you have TH, T helper lymphocytes, which is going to affect, because they are going to, T helper lymphocytes are found everywhere in our body, and they are going to help all the reactions to happen. So if this guy is targeted, there is no help and our body's immune system goes down. Okay, always remember that. Next question, 63rd. Where will you look for the sporozoites of malarial parasite? Tell me the answer for this question. Where will you look for the sporozoite of malarial parasite? RBCs of humans suffering from malaria, spleen of infected person, salivary glands of freshly molted female Anopheles mosquito, saliva of infected female Anopheles mosquito. What do you think is the answer for this question? C. Everyone says C. Are you sure about C? Yes, yes. So all of you heard me correctly, and all of you had great teachers at schools also. I think I should be giving them due credits as well. Okay, so your answer is C. The sporozoites, my dear children, you can find in the salivary glands of the molted female Anopheles mosquito. Not in the saliva. Do not go with the saliva and things like that. Okay, it's female Anopheles mosquito. Cool. Let's go ahead with the next question. 64th question. Can you try to find the answer? Children, are you sure? Are you sure you gave the correct answer for the previous question? Okay, 64th you are giving now. Chalo, let's solve 64th, and I have to ask you something, okay? AIDS is characterized by a sharp reduction in what? Sharp reduction in helper T cells. I told you already, it's T cell that is mostly affected. So it will be this, okay? It will be this helper T cell. So A can be the answer. Now, going to 63rd question. Just because Jasmine Ma'am marked this, can you believe directly without thinking about it once? Can you believe that directly? That is why I actually wanted to point it out. So there will be so many people out there teaching a lot of things, my dear children. You have to do your own research when something new comes up. No, over here the answer is, where will you look for sporozoites of the malarial parasite? RBCs though you cannot find. RBCs of human suffering from malaria, there you cannot find sporozoites. Say there will be different stages, my dear, gametocyte and things like that. Spleen of infected person, again you do not find it there. Salivary glands of freshly molted female Anopheles mosquito, do you find it there? You just know the salivary gland, it is there. I have taught you salivary glands, which is why you went with that. But I saw only one person who asked me, what do you mean by freshly molted female Anopheles mosquito? That means basically it has just been formed. So something that is being formed, something that is not infected, do you think it can transfer the disease? Do you think it can transfer the disease? No. So do you think that can be an answer for this? No. But over here, saliva of infected female Anopheles mosquito is given. Do you think that can be the answer for this? Yes, this is the answer. This is not. Please be careful with these type of things. You have to be little more vigilant. Okay, so this is your answer. Infected, it is clearly written infected. This is freshly molted, something that is not infected. Okay, ma'am, you don't, I said don't go with saliva. That's what I was just trying to confuse you guys. Okay, so please always remember, you have these terms written over here. You have to be very what do you saw? Chirag, yes, I asked it before. Okay, anyways, ultimately, tell me, playboy. Okay, ask. How, ma'am, you didn't get it? Dear, it is salivary glands of freshly molted female Anopheles mosquito. That means it has just been formed. So how do you expect it to be infected? Over here, infected is clearly mentioned. And whenever you say salivary glands, obviously salivary glands is something that will be producing saliva. And this saliva is going to get inside the human body, right? So obviously, whatever is there in salivary gland will be produced in the saliva also. So it will be there. Okay, so please go with this option, not with this option. This would be totally wrong because this particular letter is there, word is there. Okay. Here the helper T cells is correct. Ma'am, I told D first only. Okay, Abhijit, great. Now, 65th question, try this. T lymphocytes mature in dash while B lymphocytes mature in dash. Most appropriate combination of words to fill in the blanks is. Now that is easy. Oh my God, playboy. Great line, but not to be used here. Okay, chalo. Study. We have to finish, right? So someone is saying A, someone is saying B. Okay, let's see. T lymphocytes, my dear children, as the name suggests, T stands for thymus. So they are going to mature in the thymus. Okay, and basically B lymphocytes are going to mature in the bone marrow itself. Listen, bone marrow and thymus are the only primary lymphoid organs. Only primary lymphoid organs. These are the organs where three things happen. Okay, they proliferate, they mature, and they become antigen sensitive. They become what? Antigen sensitive. That means they know that, okay, this is something that is not from my body. This is something foreign. I have to react to it. But do they react over here? No. Where do they go and react? They will be going and reacting in the secondary lymphoid organs. Always remember. So they become antigen sensitive in the primary lymphoid, but they become, what do you say? They become effector lymphocytes. They start showing their effects in the secondary lymphoid organs. And can you tell me certain names of the secondary lymphoid organs? There's someone out there, secondary lymphoid organs, not secondary lymphocytes, secondary lymphoid organs. Can anyone tell me certain examples of secondary lymphoid organs, my dear children? Yes, that is how it came. T for thymus, T lymphocytes, and B for bursa fabricus, okay, bone marrow. Spleen, Peyer's patch, appendix, lymph nodes, Pavanraj, awesome. Okay, so I see Soumya, Shiva, Yogi Gowda, Trisha, Shashank, Sahanshal, Kushal, okay, Chinmay, Trisha, all of you, Akshita, Ibani, Shashank, all of you, great. Okay, so awesome children. Chalo. Now, MALT constitutes about -% of the lymphoid tissue in the body. MALT constitutes about -% of lymphoid tissues in the human body. Can you tell me? 70, 50, I see lots of different answers. What is it, A or C? Open your NCERT everyone, open your NCERT, go to human health and diseases, go to lymphoid organs, just after autoimmune diseases you will find the answer. 50, yeah. Come on children, go, go, go. Okay, all of you are saying 50? All of you saying 50? Then let's go with 50 itself. Okay, it's in page number 138. Okay, thank you Swapna. Is it more than 50%? Is it more than 50% or not? Just give me a second, okay children? Just give me a second. I'll be right back. Ayo, just a second. Yes. Okay, so it is 50, and with that we complete this particular chapter as well, and I'm going to end the live soon, but after answering to your doubts. But before that, I want to tell you something very important of this particular chapter. Listen to me carefully. Something that I have not put over here. Okay, now listen to me carefully. How many type of drugs do we have? We have opioids, we have cannabinoids, and we have coca-alkaloids. Always remember that, okay? In the case of opioids, they are going to affect your central nervous system and your gastrointestinal tract, GIT, okay? Now, the second comes after opioids is what? After opioids, you have cannabinoids. You have cannabinoids. It is going to affect your cardiovascular system. Vascular system. Okay, now third comes your coca-alkaloids. Coca-alkaloids, what does it affect? What does coca-alkaloids affect? It is also going to affect your CNS, and it's also going to mess up around your dopamine secretion. So dopamine is a happy hormone and things like that. Okay, so basically it is going to affect the dopamine receptors out there. Now, very important things. Now listen to me carefully. Okay, example of opioids is heroin and also morphine. Morphine is sedative and painkiller. Heroin is a depressant. Cannabinoids, you have hashish, ganja, marijuana and things like that. Okay, and they are also known as smack. Okay, now coca-alkaloids, it is derived from erythroxylum coca, my dear children, and basically example is your cocaine, and it is known as coke or crack. And they also lead to euphoria. Okay, they lead to euphoria, that means intense sense of happiness because dopamine receptors are being blocked, that is why you're continuously happy, and also hallucination. Apart from coca-alkaloids, do you know any other hallucinogens? Children, do you know any other hallucinogens? Can you tell me the names? If you know the plants from where you get them. Two of the things are given in your NCERT itself, and you should be knowing it. Two, Datura, exactly. Datura and Atropa belladonna. Datura and Atropa belladonna, both of them are hallucinogens. Okay, remember, apart from coca-alkaloids, you have them as well. Now, one more thing. Tell me the name of the drugs which is used to treat insomnia. That means if a person is not able to sleep, so there's a drug given to them. What is that particular drug? Any surety about that? Somnia papaver somniferum is the plant where you get opioids from. Okay. Barbiturates, exactly. So barbiturates and amphetamines, where do you need them? Barbiturates and amphetamines are needed, okay, to treat insomnia and depression. Now tell me one more thing. What is that drug that increases the blood pressure? What is that drug that increases the blood pressure? Okay. Can anyone say, Soumya? Yes, dear. Ma'am, I've completed all the chapters except molecular basis of inheritance. Please do that. Very important chapters. Adrenalina. Okay. Adrenaline is a hormone. It is released because of what? What is it? Come on, children, you can do it. Think. What is that something that increases your blood pressure? Very, very important question. Someone who, yes, great, Madiha. gave the answer. It is what? Nicotine. So nicotine is there in tobacco, and basically this nicotine, my dear children, goes and affects the the adrenaline glands and releases adrenaline and noradrenaline, increases blood pressure and things like that. Okay, please remember it. Apart from that, apart from that, one more question. What is that something that is used by the sportsmen? The sports people know they use some drug very much. What's it? What is it? Drug. I'm talking about drug. Any guesses? Cannabinoids, exactly, Akshita. It is cannabinoids. Okay, this thing that the sportsmen use, they abuse this. That means basically they use it very much. Cannabinoids. Okay. Now apart from the drugs, okay, there are certain anabolic steroids that is being used. Anabolic steroids that are being used. So what do they do to the men and the female? In the case of female, you have mood swings, irregular menstrual cycle, little masculine body and things like that, enlargement of clitoris. This is one very important thing. Okay. In the case of men, there is enlargement of prostate, there is reduction in testicle size, okay, and what? And yeah, and basically acne and every mood swings again will be there. Okay, so please make sure you know all this thing. Apart from this, I want to ask you a question. What is that particular antibody released at most amount during allergy? During allergy, what is that antibody which is released in most amount? Can you tell me? Can you tell me? IgE. IgE. What is the answer? IgE, exactly, immunoglobulin E. So IgE, allergy, both of them ends with E, you can remember like that. So IgE basically goes and binds on the mast cells. Okay, these are the mast cells, and the mast cells, when they get binded, onto which if suppose dust and any particle comes and attaches, this is the antibody onto which the antigen has come and attached. Now the mast cell becomes activated and releases histamine and serotonin, which leads to inflammation, which leads to what? Inflammation. Hence also it will lead to vascular dilation, as in, for example, your blood vessels are like this, now it will be like this, so that so many cells would come and act, and when they do that, you get a response, hyperactive response, that is known as allergy. Now tell me this thing also, what is that drug that is passed first from the mother to the baby? First, not drug, okay, I said drug, first antibody. Okay, first antibody that is passed from the mother to the baby. Can you tell me? IgG, exactly, it is what? IgG. I have to increase the more space. So it will be IgG. That is passed from placenta of the mother to the fetus, that is the baby. And from the milk, my dear children, from the milk, what do you get? IgA. So always remember, Amma. Okay, A for Amma, A for milk. So basically it is IgA that goes from the milk, and the first milk is known as colostrum. Okay, in the case of antibodies, how many how many antibodies do you have? GAMED, gamed. Remember like that, GAMED. Okay, now yes, Sanjana, benzodiazepines are also there in barbiturates and things like that. Okay, now going ahead, gamed. What else do we have? What else do we have? What else? Antibodies. Yeah, the structure of it. How many heavy chains do you have? You have 2 heavy chains, my dear children, H2, and you have 2 light chains, that is L2. So H2, L2 is there. Apart from that, do you have variable region? Yes. So this is the variable region, this is the variable region. So both of them have a variable region. Here also you have variable regions, and then the rest of this is what? Constant region. In between the heavy chains, you have sulfur bonds, disulfide bonds, like this and like this also. In between them and in between 2 of them, and in between among them also. Here also you have 1. Here also you would be having 1. Yeah, that's a double S bridge. Cool. Apart from that, was it missing? Okay, children, fast, okay? Will you answer me fast? Yeah, these are the antigen binding site. The variable region is the antigen binding site. Chalo, answer fast, okay? Tell me 5 names of bacterial diseases. Tell me 5 names of bacterial diseases. Bacterial. 5 bacterial diseases, name fast. And I hope everyone has liked the video. If you think that I taught properly, please do not forget to like the video, share it with your friends also. Okay. Pneumonia, whooping cough, okay. Ringworm disease, abba. What are you saying? Okay. Write it in once go. You take your time, write it properly. Okay, please take your time. Okay, can I say from your NCERT directly nothing apart from your NCERT? Okay, listen to me carefully. Typhoid. I am going according to the flow of NCERT. Okay, typhoid, pneumonia. In that pneumonia paragraph itself, diphtheria, okay, dysentery, and I think cholera is not been mentioned, but yeah, cholera also. Okay, and plague. Yes, plague is mentioned. So 5 things in your NCERT you have to go through it. Malaria is caused by, what I am saying, typhoid, sorry. Typhoid is caused by what? Salmonella typhi bacterium, okay. Pneumonia is caused by pneumococcus, streptococcus pneumoniae, and also Haemophilus influenzae. Now what do we have? Diphtheria and dysentery and also plague. That is separate thing. What is an example of viral disease? 2 examples of viral disease. Can you give me? Ma'am, chill. Thank you. Thank you for reminding me that. Example of this disease, viral diseases. There are 2 of them. Yes, common cold and what? One more. HIV, measles. Yeah, HIV, polio virus, HIV virus, measles, common cold. Okay. Now, common cold is caused by what? Rhinoviruses. Okay, common cold is caused by rhinoviruses. Apart from that, my dear children, apart from that, what do we have? Tell me examples of protozoan diseases. Protozoan diseases. Okay. Chalo, from now on, I am going to read the names. Okay, protozoan diseases. Chalo, let's start. Okay, Shiva, I am reading like that. Okay, malaria. Ruben, please check, Baba. Naga malaria, Bhumika malaria, Madhiha malaria, Safia amoebiasis, yes, and Akshita malaria, Pavan rhinovirus. Old. Okay, Kavya, both malaria and amoebiasis. Great. My dear children, protozoan diseases are 2 only, and one is malaria, second one is amoebiasis. Okay, malaria is caused by Plasmodium. It is caused by different species of Plasmodium. Plasmodium vivax, Plasmodium malariae, Plasmodium falciparum. Falciparum is the most malignant version of the malaria. Okay, it will be causing the most malignant version of malaria. Now we have amoebiasis caused by Ant-amoeba histolytica. Okay, Ant-amoeba histolytica. It is spread through food and water and contaminated things like that. Okay, now always remember the female Anopheles mosquito is transferring the malarial parasite as well. Now what do we have? Okay, 2 diseases which are caused by Aschelminthus. 2 diseases which are caused by Aschelminthus. Ma'am, your name please. You do not know my name. My name is Jasmine Joy. Elephantiasis, Ascariasis, exactly. Ascariasis is caused by Ascaris. Elephantiasis is caused by filarial worm, Wuchereria bancrofti, Wuchereria malai. Okay, always remember they are going to affect your lymphatic vessels. Okay, Wuchereria, that is elephantiasis, that is why you have a big leg over there, because it is affecting the lymphatic vessels of your lower limb and the genital organs as well. Okay, so that is this, and it is spread by the bite of Culex mosquito. Wuchereria bancrofti, Wuchereria malai, they spread through Culex mosquito bites. The name of the mosquito is not given in NCERT, so it won't be coming, but please know that. And in Ascariasis, the most important symptom that you get is blockage of intestine. Okay, intestine becomes blocked. Soumya, thank you so much. Okay, oh my God. Okay, next question is, can you give me example of fungal disease? Can someone give me example of fungal disease? Ringworm, exactly. It is caused by 3 groups of fungus. Trichophyton, epidermophyton, they are fighting. Okay, epidermophyton, trichophyton, and also microsporum. Works. So that is done. Human health and diseases. Now one more thing. Basically, what is that? Something that causes fever and chills in the case of malaria. So basically, when I say RBC, in the RBC, gametocytes are being produced. In the RBC, gametocytes are being produced. And also, you know what? The Plasmodium is feeding on our hemoglobin. It feeds on our hemoglobin and breaks it down into heme and globin. This heme is very toxic for the Plasmodium. This heme is very toxic for the Plasmodium. So it converts into something else that is known as hemozoin. But until now, it was there in the RBC. Once the RBC bursts open, what will happen? The hemozoin is released. The hemozoin is released, and this hemozoin will cause fever and chills. Okay? It will cause fever and chills. Nikhat, thank you dear. for reminding me to drink water. Okay, I'll drink. Okay, apart from that, cancer, my dear children, you have different causes. Oncogenic viruses, proto-oncogenes, radiations, ionizing and non-ionizing radiations, and chemical carcinogens. Okay, works. And cancer related question, yes, I'm coming to that. Chemistry live Chinmay, it will be there tomorrow. Okay, where is it? Okay, cancer related question, what are the features, what is not the feature of cancerous cells, that might be asked. So loss of contact inhibition, this is not a feature. So basically, presence of contact inhibition. Suppose the cell is still showing contact inhibition, that means it is not a feature of cancerous cell. Cancerous cell has lost contact inhibition, means suppose the cells are dividing, they stop division when they come in contact, that is known as contact inhibition. In the case of cancerous cells, they have lost contact inhibition, my dear children. Now second thing, metastasis, they move from one area to the other area, so they spread. Okay, now one thing, in the case of cancer people, you give biological modifiers to your body, immune modifiers. What do you call that modifiers as? Ma'am, tomorrow class timing, I think it will be there in the morning hour. What do you call chemistry? If you're talking about biology, tomorrow biology is not there. Introns, Safia, Baba. When someone is having cancer, they are given some immune modifiers, and you call that alpha interferons. Okay, you call it alpha interferons, different from the other interferons that you spoke about initially in the start of the chapter. Okay, physics will be there, I think around 12 or something like that. Okay, works. Now anything else that I want to say? I don't think is there. Okay, yes, I think that's all. Do you have any doubts, or shall we end the class, my dear children? Enough. Okay, ma'am, full hard work. So children, I will be making a video. The second part of the video will not be live, instead it will be recorded and it will be published day after tomorrow. That is on your day of your exam, on your chemistry exam and your physics exam, and I hope you are going to study well, and please go through each and every questions properly, go check it out, don't just believe me and just randomly go like that. No, you should be checking it out by your own sense, and yes, I will be helping you. If you have any doubts, you can put it down in the comment section, and please let me know how was the class in the comment section below, and also share it with your friends if you think it was helpful. Okay, so Chinmay, thank you so much, dear. Ma'am, opioids are opium, that is heroin and morphine, my dear children, and I have explained about that. Can you please check it, iClown? Okay, Nisha, thank you so much. Thank you, dear. Pavan, thank you. Theo, Harshita, it will be there in the part 2. Okay, all the other important chapters of PU2 and PU1 will be there. And Chirag, 36th question. Yes, we were stuck in the 36th question. They can't. Okay, thank you everyone. Okay, just a second. Okay, for children who want to leave, please leave. Let me go ahead with this question that we had. 36th question was there. Yes, this one, right? I will check this and I'll let you know. Will that work? I don't wanna let's say make it difficult for you or give wrong information. So I'll check it and I'll give you in the comment section below. 36th question, the final answer. Because it is kind of confusing. The first meiotic division during oogenesis is completed at the stage of. Okay, it will be this, secondary oocyte only, not this one. Not this one, it will be this. It will be completed at this stage. Okay, first meiotic division during oogenesis is completed, that is being asked, so it will be secondary oocyte. Okay, and it will be within tertiary follicle. I think that is the correct answer. Okay, but still I will be what? Confirming this once more. Okay, now 65. Okay, 60th question now. Okay, children, all of you others, please, you can leave, okay? And if you are done, I will be just solving the doubts of the children out there. Natural killer lymphocytes are an example of natural killer cells. They are a certain type of cells, right? So it will come under cellular barriers. Okay, cellular barriers as in the cells that will be helping us protect. It's not specific. They are not like I will be killing only these cells, I will be working like this, nothing like that. So natural killer cells are the one that actually kills the virus infected cells, the cancerous cells and things like that. And apart from natural killer cells, you have neutrophils, which are phagocytes, which takes in the cells and kills it inside. Similarly, you have macrophages. Okay, you have monocytes also. PMNL is what you call as neutrophils. Okay, polymorphonucleoleucocyte, because they have a multiple lobes in their nucleus. That is why. Rutvik, ma'am, is this the next class? Last class? There will be one more class. I think it will be recorded. 65th question. Srinivas, you're most welcome, child. And thank you so much, children. I was not able to read a lot of your answers, but yeah. T lymphocytes, yeah, this one, 65th question, Nagaratnamma. Okay, T lymphocytes mature in the thymus. So you have two regions. Thymus is over here in the central region behind the sternum. Sternum is a chest bone that you have on which the ribs are attached, right? So behind that you have thymus. So thymus is a place where basically the T lymphocytes which are made in the bone marrow will come and mature. Okay, similarly, bone marrow is a place where all the cells are formed, and B lymphocytes stay there and they get matured there itself. They do not move around here and there. Okay, Anaga Moose, yeah, Malayali, I'm here. Another class we will, ma'am, we need live class. We'll definitely think of that. Yes, thank you, dear. Okay, so children, done with that. All the 66 questions are done, I believe. Shall we end the class? Thank you. Chalo. Chalo, then let's go home. I also want to go home, you also want to go home. Right, so ma'am, dinner, we would be having dinner. Ma'am, shall I take BTech Biotechnology? Is it good? Depends in which country you are living in. For India, I won't actually suggest with going with that, but yeah, since it is developing, we can try. Depends on the good colleges that you get into. Okay, that's an honest opinion. So that's all, children. I hope all of you have liked. Okay, please like the video and go off and also comment down your valuable things. So that's all. Wishing you all the best, lots of love, and next class timing will be updated soon in the WhatsApp channel. So please join the WhatsApp channel. Okay, first PU bio will also be there in the next video. Bye bye, take care, lots of love, children.